Dr. Lorenza Fernandez is from Spain. She has been working with Aerosol Dynamics Incorporation for many years, and they have developed different types of instruments based on the conversational growth technology for different applications. So please join me in welcoming Dr. Lorenza Aguirre and Fernandez.
Thank you. We developed what is called the water-based conversational growth technology, and me coming from UCLA many years ago, working with toxicologists, immunologists, exposure assessment people. I got a chance to learn what they showed, some of the technology that was currently available. So I started working with my colleagues to see how we could use this new technology to improve the assessment of breast health outcomes related to exposure to identifying. So around 2014, we developed this technology that is a moderate and flow condensation, and uses water to grow particles as small as 5 nanometers to a range around several microns that makes us easy to collect them, count them, deliver them. So it's a really versatile technology that allow us to play with those ambient particles, especially the smaller sizes that are really hard to collect and work. So the technology consists of three parts. It will be what we call the conditioner, that is a cold section. The initiator, that is a small section that is hot, where the supersaturation of the water happens, and where the particles start growing slowly. And the moderator, that is a cold last stage, where the particles continue growing until they reach the desired height. But until this part allows us to control the relative humidity of the exit flow. So that control here will allow us to do dry deposition, wet deposition, cell delivery, and many activities. So by controlling not only this temperature difference between the conditioner and the initiator, with the flow, we see on the right side plot how big the particles can grow depending on what is the flow over a certain length of this tubing. So we can deliver particles from around 2 microns to 4 microns, 6 microns. So we can grow particles from 2 to 6 microns. We've been working now with our group in Baltimore that wanted to do some MEMS after collecting the particles in emulsion, and we are able to grow the particles up to 2 microns droplet size. So having these particles in this coming out in a pretty big size range allow us to so it will allow us to collect the particles as small spots of dry particles that can be used to do chemistry. You can do also TM, what our previous presented. So, and we can also collect it into liquid as suspensions, some suspensions of terms of interface. So you can see the particle and the water soluble material that is attached to them. So based on this technology, we develop new implementation to help us to figure out the toxicological properties of airborne particles and also identify infections by viruses. So the 3 technologies that I'm going to talk to you about right now will be what we developed, the online monitoring for the oxidative stress system that we call it OMOCA, and basically will give an idea of how capable are these airborne particles to generate reactive oxygen species. We have the direct in vitro exposure type and dosimetric aerosol in vitro inhalation device, David, that's systems that allow us to do in vitro studies for toxicology. And the last system that is the BioCascade and the Vivas. You've seen the Vivas several times over this presentation on the back of the book. And the addition to that system is our BioCascade that will allow us to do live transformation collection of the bioaerosols. So what we started with the OMOCA is when I was at UCLA, it was this big move about how the generation of oxidative stress was a major concern because it was one of the ways that were associated with particles to health-related issues. A lot of them we have Alzheimer, we have the asthma and COPD. We also have cardiac, heart-related issues. So all these health effects were just coming like, oh, oxidative stress is going to be really important. So when we were there, they thought that this pathway was through the generation of reactive oxygen species to the different components associated with thermal pathway. So while at UCLA, we developed a new assay that was called the type 3 assay, and it's a chemical-based assay. So there are no cells involved, which made it pretty easy. And it's been widely used right now in our research area that we identify some of these toxicological components. So this assay does measure the capacity to generate aggregates through this transfer of electrons from DDT. And the activity is expressed at the rate of consumption of this DDT per microgram of sample or per air sample volume. So if you know the mass that you are using for this assay, you can do it in micrograms. Prefer to do by mass because it's easier to know that a person how many cubic meters of air they will breathe a day and how much mass we are on our intake. So the system consists of our liquid spot sampler, that is a smaller version of the Vivas. We collect the sample on a small volume and then around 0.5 emails. And then we transfer it to the chemical module that we developed. So we have a reaction chamber where the nitrogen reactor is reacting with our particles, and then it's sent to our detector that is going to monitor the intensity of that reaction. With this assay, we can collect kind of near real-time on a system that we deployed and just forget about it. And in this case, for our testing, we collected samples in Berkeley, now our window, and we have 3-hour samples. So in this case, we compare our DDT results with PM2.5 and black carbon that was monitored on a station about a couple of miles from where we were. So what we saw is that we have considerably diurnal variability on the oxidative potential of these particles to generate these aerosols. And in this case, we also saw some correlations with the black carbon and some of the PM mass, PM2.5 mass in our neighborhood. I have to say that our lab is close by to major freeways and roads, so we are basically impacted by primary emissions from vehicles. So this system will give you semi-real time down to, in this case, 3 hours information of this oxidative capacity of the samples that could be later on related to some other. So the next system is the DIVE and David. So we all know how hard it's always been to do in vitro studies with particles collected on filters. We need to extract it, we need to pre-concentrate it, we need to give them. So there's always been issues with dose and dose response characteristics. So we used our technology to deliver particles directly onto the cell. We don't need to do molecular extraction and we prep them. And we deliver the particle gently over the surface of the cell, the in vitro system. So we try to keep the viability of those cells and not generate the stress that can keep us some fault of resource. So the first system we developed was what we call DIVE. That was a system developed to expose tumor cells at that time, 10 years ago. It used to be the most common in vitro system to run. So on this system, we have the conditioner, the cold, the hot, and then we have a little nasal temperature to keep a range. We created a little chamber that will keep our cells at 37 degrees C, and we maintain the average of the incoming flow at 95% average. What we found out is that during sampling, if we don't add CO2 to our main flow, the seed medium also retains the pH, but not good for the cell. So by adding this extra inlet of CO2, we solve that problem, and we were able to use this system for several years. So following up the system, and as the years pass by, and people start using what we call the ALEX system, the early green interface cells, that they represent or mimic better what's happening in our labs. We bring these systems to what is the date. So we also have the conditioner, the initiator, the moderator with the third stage to better control our temperature, because these ALEX cells are more sensitive to changes on average. And then we create our chamber to hold these ALEX cells. So the first time we took dive to the field was in Michigan. And we exposed cells, VS2B cells, for 3 hours to both indoor and outdoor air. And we measured both IL-8 and IL-6 release from the cells that were exposed. And what we can see is that our controls were really happy on the system. And then we start seeing considerable changes of the IL-6 and IL-8 release when they were exposed to ambient particles. And there seems to be a significant diurnal pattern. We were close to the parking lot in the university, so we see that in the mornings when it was cold and the cars were running to the parking lot, we used to have higher release of these markers that we will have in the afternoon. The same thing for the IL. So this system was our first step to do some direct cell exposure to ambient particles with a portable system that would allow us 3 hours exposure time resolution. Then we moved to David, and most of this work with David has been done with CWES group and in Caseville, you know, the Caseville. And this system was developed to use with ALEX system. So the way that these cells work is they have the feeding medium underneath and they are exposed to the air. So working with these cells were more complicated when I was working in a different system. So the first thing we did is we developed by a 3D printer these membranes that were capable of holding these cells under our current system. But afterward, we found it was really complicated to do this and was not going to be useful for other users to have this 3D printed involved. So we modify our collection chamber to keep the cells on the corner transport plates that basically covers the membrane. So now you just find the plates, have your cells, and then just move it on. So the first step we did with this system is make sure that we are able to control the dose that we are giving to those. So we get some SMPSs, APSs, we estimated the particle mass that was going into our level system and how much mass was deposited on our different grids. And you can see that we have a pretty good correlation over time. So if we know the mass that we are exposing them to on the inlet, we can estimate how much mass we are going to be. There's a really nice work to characterize excellently all these properties by Ezra Priya that should be coming out soon, hopefully. And if you're interested to see how you can control the dose using this system, that will be available pretty soon. So the second thing we did is these cells are pretty tricky to work with, and they are not really happy if the conditions change. We did some cell viability studies, changing the different flow rates that we were exposed to. We have 6 liters per minute unit and around 4 liters per minute. So we see that up to 20, we can increase in further study later on up to 30 or 40 minutes. The cells need to be pretty happy with the system. So we are not going to generate stress that is going to affect our results from the exposure to particles, nanomaterials, bioaerosols, or 1%. So the first test with this system was using silicon oxide and carbon oxide nanoparticles. This is some cell microscopy images from the control cells, the cells deposition of silicon oxide in the cells, and the carbon oxide from the cells. So in this study, we saw we did two different tests for viability, the LDH and the DB, and we can see that in general, the silicon oxide is not toxic, but the carbon oxide has high toxicity on the cell's viability with doses up to 10 micrograms. And I want to say that these doses of micrograms percentages were worth in really short exposure times. So we can achieve these high concentrations in really 10 and 15 minutes of exposure. So then we look at the release of vanillate as a marker of toxicity, and we can see that the silicon oxide, unless you go to really high concentration, you are not going to see any toxicity. While the carbon oxide, you start seeing an increase in the release, even at low concentration. So overall, there was a test to figure out if the results of the system were similar to what other researchers have found in the literature, and yeah, it's confirmed that we can get the same result in shorter period of time with better tox control. The following study was done using more realistic conditions with some negative fumes. And what we did is we generated fumes on a welding in our Dr. Silva's lab, and then we used the interexposed cells. We did two conditions. One was the regular welding fumes, and the other one was using this compound, the tetramethylxylene, that is usually added to reduce the toxicity of the emissions from the welding. And what we saw is that when we have this TMS added to the welding, the toxicity, in this case measured as cell viability, was not that high, so we only lost about 10% of the cells, well, up to 8 minutes exposure time. While we don't have that controlling compound, the toxicity is much higher than any given exposure time. And finally, we have the biocascade vivas. So that the vivas, the original type was kind of the grandfather colorway of the vivas, but then it was slowly modified to collect bioaerosols. We needed to act them smoothly on the surface, and with Dr. Silva and his team, we modified that system to become what is the corvios. I won't go give us used it enough. But one of the problems is that, yeah, we know that they are bioaerosols. You know, with COVID, we found that we know it's there, but we still don't know the size. And a lot of the infectious disease people, I think that's people see what is the side effect and what that might start to do measure control measurements, use protective equipment. So we came up with this idea of what we call the biocascade. So basically, it's a sequential impactor that will allow us to collect viable bioaerosols with 3 different fractions. One fraction will be less than 10 micron particles. The second one will be 4 to 10 microns, and the last stage will be 1 to 4 microns. Anything smaller than 1 micron will be then sampled to another system. In this case, we hook it up to the vivas, but you can put a filter, you can put any other like gel filters, you can put any other collection system at the end. So the original biocascade, we did some testing. We were able to get the around 9 micron for the first stage, around 4 on the second stage, and 1.5 microns on our last stage. So some of the configurations, this is how the biocascade looks. This is hooked up to the original vivas, the prototype, and Dr. Silva will lab. Some other ways to use in this system, like Romeo comes in Baltimore, that is using our biocascade collector with one of the commercial vivas to collect the cell aerosol to monitor influenza or COVID. So some of the studies have been ongoing with the Silva's group, especially with PRIA, with his PhD student with the group of other students, they've been monitoring COVID. And they've been doing this latest study that they took the system of the device and the biocascade to a single family home. And they've been collecting samples for 30 minutes and measuring the presence of the SARS-CoV-2. And something that it was shocking at the beginning, I don't know how far they've gone with the experiments, is that they were able to find SARS-CoV-2 on the stage 1. And the stage 1, remember, is the largest particle fraction. You are going above 10 microns. So that's one of the shocking points. I don't know, I will talk to Shukri and see how well it's doing with this stuff. And the latest is they changed on the bioscale instead of having wearable collection. They put cell in vitro system collecting the particles directly onto the cells. They use they wanted this system to monitor this human coronavirus that seems to be seasonal, and they use red cell to do the export. So what we have here is the particle feeder cell, how they look, and the incubator control. And they found on the stage 3 of the biocascade, the one between 1.5 and 4, and the 4th stage, that is the vivus, less than 1.5. They found that they could collect viable coronavirus. In this case, we have the 4th stage that was incubated, and we have 3 days post inoculation, which is starting to do damage higher, and 7 days for inoculation, basically. It's been considered on limits. So using the biocascade, they're also able to do cell studies with that. And finally, they've done some Excel sampling too, and they seem to be pretty happy with this unit. So that's another of the possibilities that we have to do this biocascade with us. And finally, all the collaborators, I'm sure I'm missing some, especially from Teamwork team, because there are so many students that have been working with that. A lot of the grant supporting, basically, we get funding from SBIRs. And finally, I'll link a few of the papers if you want to follow this work that I haven't read. So there's been a lot more going on with these. Thank you.
Thank you very much for the wonderful presentation from a business perspective. Next, we are going into the third presentation, and very often we would consider the inhalation exposure related to the respiratory system, but Professor Naresh Kumar actually is working on exposure through the eyes, a very different route. So please join me in welcoming Professor Kumar for the presentation on this eye exposure.
Thank you. Great, thank you. So this work is just not about eye, it's a work of the evolution of my work on air pollution and human health for the past 20 years or so. So pardon me, I have different material, but it will provide you some glimpses of how my research has evolved over time on air pollution and health. So before we get started, I would like to declare I have no conflict of interest in the motivation and conduct of this research, the collection, analysis, and interpretation. This work has been funded by several of the NIH grants, also some EPA grants, which I have not listed here. Okay, so why do we care about the air pollution in human health? As you know, about 400 premature deaths. That's one of the estimates by WHO, and of these 90% are in low-income countries. Now, there's also another thing, especially in the high-income countries. When we think about ambient air pollution, what has happened? The trend has been declining. When I map the US EPA data for the past 25 years or so, there has been a substantial decline in all the ambient criteria pollutants. What's happening to asthma, COPD, and other cardiopulmonary disease that is going on? So I call this the air pollution health paradox. It doesn't mean that if there's a decline in ambient air pollution does not impact cardiopulmonary disease. The fact is we spend more than 90% of our time indoors, and indoor air pollution is one of the big culprits. Okay, so secondly, there is very limited data on indoor air pollution. If I ask you what is the aerosol concentration of different site distribution in your home, perhaps you won't be able to tell me, or perhaps there are no legislation like the National Ambient Air Quality Standards legislate for indoor air pollution. Only the OSHA does regulate the air pollution in the indoor environment in occupational setting. So in home environment, we have little to no understanding. So throughout this presentation, I'm going to focus on indoor versus outdoor and some of the new research that we've been conducting on how to monitor in real time some of the indoor air pollution and quantify their associated health risks. So Dr. Fernandez was saying the air pollution does affect a variety of disease outcome. Until very recently, we have been focusing on largely the cardiovascular disease, but that's not the case. As you know, there are different signs, morphology, chemical composition, characteristics, all these things do dictate different types of response, different organs. So some of my research does suggest that the even the aerosols can be delivered into brain, into lungs, into heart, plus our eyes are also already exposed to air pollution, so they have direct access. But that's not all. As I was saying, climate does impact weather, and weather does dictate the aerosol size and type distribution, their fate and transport. Now, there are other factors that are present here as well. For example, somebody is exposed, have comorbidities, somebody who is elderly versus child. There are a host of other factors who impact health and well-being. So for me, when we study air pollution, we should be focusing on systemic toxicity, not just one organ or just one cell line or just one type of experiment. First, when you think about air pollution, they do not exist in isolation. So another focus on my research will be on to study their impact with the mixture of air pollutants. So air pollution have direct impact, weather conditions have direct impact as well as indirect impact. An example I'm giving you here. For example, meteorological condition. As you know, we have very cold temperature for the past 7 days or so, 3 days or so. You might be seeing a lot more frequent ER visit due to asthma and respiration. It's largely because of drop in the temperature. On the other hand, if you think about your indoor air pollution at home, it must have been increased. Sheer fact is because your air conditioning system is not taking out excessive moisture, as a result, some of these particles will begin to stay airborne for a longer period of time. So meteorological conditions also mediate the health effects of aerosols. That's the second aspect. So what I'm going to cover in today's talk are these following topics. Examine the relationship between indoor and outdoor PM in low and high income countries. Then describe the health effects of indoor PM. And finally, present some of the examples of real-time air pollution surveillance and their associated health impact, including its impact on the human eyes, as well as asthma. Just to provide an overview of the summary of some of these traditional methods that we're using. These tools were deployed in 2007, 2008 in different parts of the world, including Delhi, Cleveland. So we have these carbon impactors. We have this is photo-based aerostat. Then we have passive sampler, and they are different cyclometers, PM2.5, PM10. Then we also have some real-time instruments too, and all of these instruments were deployed simultaneously indoor as well as outdoor. Now, as I was saying earlier, when you think about the health effects of aerosols, when you think about the size distribution, because size will dictate where the aerosols will be lower. As you can see, this is a real world sample collected from Delhi. And many of these, when you think about PM2.5, it means anything smaller than 2.5 micron in aerodynamic diameter. And if the sample is collected at an industrial site, many of them will be in the nano ranges. As you know, PM2.5, PM10, these are two set numbers we've been seeing. Historically, they were designed to study because PM10 is a respirable particle. PM2.5, a vast fraction of this can be delivered your heart, because they directly have access to alveoli, and this is where you have exchange of carbon dioxide and oxygen. So once it becomes part of the circulatory system, think about the particles which are 108 nanometer. Now, this can be directly delivered to your brain. So in other words, once it's circulatory, it can be virtually delivered in most part of the brain, and brain is one of the quickest blood-brain barrier, we call it, because many of the things, even in nano ranges, cannot penetrate. That's why when we design special drugs, we use nanoparticles for delivery of drugs in the brain. Second aspect, all aerosols are not the same. For example, what you're seeing, even the pollen, we are famous in Miami, by the way. I'm going to show you some slides later on. Here are example of mineral particles versus pollens. Now, this is the maple and this is ragweed. In Miami, you might be seeing oak already outdoor. As we are going to February, you will be seeing a lot more. But think about this scanning electron microscope image of ragweed. These are small spikes. If I ask you, will maple be more toxic versus ragweed? Ragweed has these small spikes. Anytime they land and you close your nose, it will deliver the allergenic protein right away, and you will begin to sneeze. You will see that sometime in April and May in Miami area. Now, but of course, these are very labor-intensive images, and you need a sophisticated instrument like SCM. But we don't have that fortunately to collect a large number of samples. However, we can do something more inexpensive. It's simple imaging of an aerosol that we collected in Miami area. Now, this without any staining, but here this is fluorescence. As you can see, all these pollens do fluoresce very nicely when you're using red light. But you can also use some stains. So we have been collecting these samples, regular aerosol contact, regular also deploying media. We can grow the culture. These samples are deployed in College Library, one of these sites at the University of Miami Medical Campus. As you can see, these are green color. These are mold spores, fungi, and also airborne bacteria. They are outdoor. Okay. Likewise, last year in February 27th, these are the pollens collected on the Coral campus. These are without any stain, and these are in red light. As you can see, you can clearly identify what type of particles are present. But the point I'm trying to make here is, in real world, you are not exposed to this pollen, mold, and other particles. It's just a kind of soup that we are exposed to. Now, another example, mass of particles. As you're seeing these filters, these were deployed in Delhi for 48 to 72 hours. The last one is the field blank, and rest are pitch blank. Now imagine a scenario: if you're in Delhi today, or if you're in China today in Beijing, or if you're in other polluted city like Tehran, your lungs will be coated like this. Of course, the mechanism won't be the same because you're breathing out two simultaneously. Filter, on the other hand, keep collecting, right? Okay, so but nonetheless, the concentration is very high. Second part is the chemical species. So you have mass, the size, your distribution. What chemical species? In case of Delhi, we saw even the very high concentration of land mass in the elemental species of these wine vertebrate matter. Now, going to the toxicity. So why don't run an experiment just taking a very selective type of particle, rather use real world particle? In this experiment I'm showing, there are different biological mechanisms that are initiated in order to study the toxicity. First, we just simply examine morphology. Dr. Fernandez showed some of the examples, but here these are fluorescence images. The blue one is the nucleus. So this is control. When we expose the real world, the cell line to neuroblastoma, the brain cells, at 15 microgram, all the nuclei, most of the nuclei are gone. Cells are still intact. So they're really struggling. Increase the dose to 75 microgram cubic meter of real world PM1 particles. These are multi-stage cascading factors. We collect these particles, extract them, and control the dose. And then here you are seeing 150 microgram cubic meter. You might ask, why am I using these P doses? This is the dose. This is the normal concentration that you see in high-income countries. In the US, threshold is 12 microgram cubic meter in any given year. In Delhi, you are going to see the concentration as high as 150 microgram cubic meter. In winter time, in summertime, that's what. Now, as you can see, as the doses are increasing, morphology changed. As morphology changing, of course, you will be also seeing other biomarkers too. LDH leakages as a matter of cell biology. And then also, we observe several of the gene expressions that are implicated in neurodegenerative disease, including Parkinson's. They were also regulated when you're exposed to these real-world particles. Another example, where we expose these cell lines to the heat as well as real-world particles. You'll be surprised to see that. In the control, 37-degree regular temperature used for culturing the cell. 40 degrees, let's assume in any given day, your temperature exposure is increased by another 3 degrees. What happens? When these two things are combined, heat stress particle, their adverse impact does increase. It happens with regard to LDH releases as well as most of the genes that are implicated in neurodegenerative disease, they were also upregulated. With regard to eye, that Dr. was saying, I also conduct a substantial amount of research. The aerosols do interact with humidity and they work in very different mechanisms. One of the target mechanisms is when you have coarse particle, they lead to abrasion. As I was saying, the morphology. If you have crystally particles, if you go to the beach, rub your eye, then that's the kind of injury. It's a very physical injury. To repair, of course, there will be other cascades of biomarkers to be initiated, like inflammation and also oxidative stress. Now, to provide you some of the data, here's a comparison of indoor and outdoor PM2.5 and PM10 in Delhi. As you can see, virtually there is no difference in the indoor and outdoor air pollution in Delhi. There is some difference in coarse particle PM10. What about Iowa? Virtually there is no relationship between indoor and outdoor air pollution. So meaning, when you want to see what's the health effects of airborne particles in low-income countries, you can still use some ambient data to quantify the health risk. In case of high-income countries, you really cannot, because indoor air pollution is substantially different compared to outdoor air pollution, especially in the high-income environment. Now, what people are exposed to in the real world. So we have been conducting another research in Baltimore and Miami. As you can see, we deploy sample outdoor, use very simple aerosol impactor. These are the particle concentration people are exposed to in real time. There is no cut area. Just anything that's airborne is being trapped. Of course, a substantial fraction is in 10. These are the type of concentration you see outdoor. And you can see there is substantial difference. So meaning the point I'm trying to make is our indoor air is much cleaner compared to outdoor air. So this is the kind of sampling setup in Baltimore area. Now, so there are two different types. One is a snapshot. Dr. Fernandez was saying you look at one point in time, you take measurement for half an hour, 15 minutes, or 30 minutes. You can use some of these data to quantify, but that does not represent real world exposure of people. So I focus substantial research on this one snapshot, and then there was health effects, including tuberculosis, including ocular disease, including again another disease. But here in this case, the fine particles instrumented by heat stress that does induces, for example, ocular surface score, that is a measure of dry eye severity, measure of inflammation, tear production. Now, we also did something more, mediating the indoor microbial concentration. One of the feature tests I was showing, we collected simultaneously indoor and outdoor. When you see this higher concentration by the microbial concentration, it leads to epithelial disruption in your eyes and also lower eyelid meibomian gland dysfunction. Now, that does not tell you what was your exposure today, what will be your exposure on Friday. That's why we have shift our gears to real-time sampling. This is an example of asthma. I am showing you here two different instruments, two sets of different instruments. This is an early sense that does measure physiological measures in real time. Heart rate, respiratory rate, sleep pattern, and it's the contactless sensor you put under the mattress. Anytime you're moving, it will breaks down your movement. How many minutes? Oh, okay, sorry. So, and these are a suite of real-time sensors here developed that monitors gases and particles of different sizes, and they are also deployed simultaneously in people's homes. So we have real-time data every 5 minutes from these instruments, and they also store these data on board. And what we see, for example, in this case, there are two different children, two different children. One has rhinoceros symptoms, another does not. As you think about this respiratory pattern, as you're sleeping, you can detect them the movement. The one with no rhinoceros symptom has a more smooth pattern. The one which has rhinoceros, as you are seeing, very abrupt. So the point is, when we try to link it with the real-time air pollution data, what's happening to these changes in particle concentration? The hypothesis here is, as the air pollution is changing, it slowly begins to impact your respiratory rate, your heart rate, before you begin to show up more severe symptoms and land up in ER. So that's the work in progress we are focusing on. Here's another example in which I show two different kids. One has a controlled asthma, and the instrument is deployed continuously for 1 full year. June 1, 2020, July 1, 2020. These are flipped. The one which has a controlled asthma, as you can see, the concentrations are very high in this child's home. Indoor average concentration 38.5 microgram per meter versus 3.7 microgram. So you can see asthmatic versus the well-controlled asthma, non-controlled asthma. The question now will be, which are those peaks that led to exacerbation of asthma? Another example about the ocular disease, these sensors deploying in people's home, only the air pollution sensor in this case, and these patients do report symptoms every week, whether you had any worsening of eye symptoms. As you can see, for the past 12 weeks, this patient with the red line reported 6 symptoms of severe dry eyes, irritation, pain in the eyes, and as you can see, he had pretty high concentration of indoor air pollution. Now, no symptoms. This person did not report any symptoms for the past 6 months. And his air pollution overall is lower than this one. As you can see, he doesn't have any peaks. And likely, anytime these peaks are spiking, maybe due to cleaning, maybe the other thing, because when you cook in home, these small particles that are released, they do not settle very easily unless you have some sufficient mechanisms to control and exhaust these emissions. So in summary, there is less difference in indoor and outdoor air pollution in low-income countries, so you can use outdoor data to study human health effects. In high-income countries, that's not the case. You need data on the indoor air pollution monitoring. So the sensors are not thirdly. Now, when we think about the indoor air pollution, it's just not a one snapshot. The sensor you design maybe can replace your fire alarm. Not only does it monitor the smoke, CO, carbon monoxide, but also monitors a variety of these pollutants and transmit these data in real time, and perhaps display to people when to take action, when to run exhaust fan, when to run open windows and doors. Because we are seeing tremendous diurnal variations. When I plot these measurements, in the evening, in the morning, you will be seeing some spike, and they last for a couple of hours to 3 hours. Now, if after cooking, let's say you open doors and windows, those spikes will grow. So there are tremendous diurnal variations, and we need to focus on what those spikes, which results into the risk of any acute effects, such as either eye or exacerbation of asthma. So high indoor air pollution is also associated with multiple adverse health outcomes. So we instead of just focusing on one disease, we need to focus on multiple disease. There is overwhelming literature that suggests that people who are living in a high pollution environment, they are also more likely to have neurodegenerative disease. So we need real-time monitoring to predict the adverse health effects. So ultimately, I would like to know if my child is exposed today, how many days in advance can I predict this child's asthma attack? So I can engage these patients in intervention strategies. Maybe begin medication, maybe use exhaust, maybe open doors and windows. So in other words, either mitigate your exposure or take certain other measures like medicinal therapies to mitigate or avoid such episodes. So there are some of the key references of the research that we have published in case you're more interested in. But nonetheless, I would like to thank you all for listening to this presentation.
Thank you very much for a very interesting comparison of indoor and outdoor. Next, we would like to proceed with the panel discussion. I would like to invite the panelists coming on board. You can have questions from the audience. Okay, I wanted to run a little bit different from yesterday. So I'm going to ask the audience from my Zoom. Is there anyone from the Zoom with questions? Okay, well, you can type in the questions and I'll read it later on. Now, getting back to the in-person audience over here. Anyone has comment, questions?
Question for Dr. Monk. Do you think we can generalize it all and say that indoor air quality is better in some type of structure versus others, like residential or versus commercial versus office, or say in the southeast US versus northeast or western, or is it just totally dominated by the building to building variability?
Well, both things do play a role, region as well as the building structure. You're on this campus here, perhaps you'll be happy to breathe one block, Victor Library, and virtual breathing, you'll be surprised. Some of the sample I showed you, you'll be breathing those. With regard to the region, yes. For example, up north, you will have in the winter time a lot higher concentration of air pollution, largely mediated by meteorological conditions compared to Miami. For example, one of the studies that I published in American Journal of Ophthalmology has shown the dry eye symptoms do peak in Miami area in March and May, and February and March. I mean, think about the dry eye symptoms in Baltimore. They do peak. I spoke about the other area. In Baltimore area, it is February and March. In Miami, it is April and May. How often so? Because we don't have pollens in Baltimore in February and March. And what's happening is the indoor exposure, because we are in heat and you don't have sufficient moisture. When you have dry air, your doctor will tell you, hey, use a humidifier. Here in Miami, you say use humidifier. That's not the case because we have already too high of moisture. So you will be saying use dehumidifier because now we have a lot higher concentration of particulate concentration. So the mechanisms, the exposures are different. So it's building specific as well as region specific. And I was telling you earlier, meteorological conditions, indoor as well as outdoor, greatly mediate the health effects of health. Does that answer the question? There's one question.
I have a question for the first speaker. Thank you very much for the very informative talk. In one of your slides, you mentioned there is a device to quantify the concentration of different carbonation species, like the single wall carbon tube, multi wall carbon tube, graphite, and the sloop, or either dust particles. So can you elaborate like what kind of mechanism you can differentiate the species, even for the say the same type of chemical, say the carbon tube, they have different diameters or length, like how you quantify the mass concentration. Thank you.
I think it has issues. The microphone doesn't work.
That's okay. All right, so the question is, can you elaborate on how we do these molecularly specific measurements of some of these aerosols and quantification and so that treatment. So in terms of being able to measure air concentrations, we're collecting these in batches of 2 minutes, 5 minutes, that's predetermined. And then we irradiate the sample with a Raman laser, that particular instrument. Then that and we collect the Raman spectra. Now the Raman spectra is it's a vibration spectroscopy technique, and it is each material has a very specific signature, chemical signature. And using that signature, you can distinguish. It's essentially what you're really measuring is a abundance of a particular vibrational group in that material. Now, even if, for example, a single wall carbon nanotube versus graphene, they both have different spectra. Even they are pure carbon compounds or materials, because of the structure of the atoms, the way they are arranged. In a single wall carbon nanotubes, there's something called radial bleeding mode, which appears in your Raman spectra that allows you to specifically, that only appears in the single wall carbon nanotube spectra, as opposed to the graphene spectra. And so you're able to kind of selectively identify materials based on their signatures. Now, I don't want to give an impression that this is absolutely foolproof. There's always 100% selective detection. There are always matrix effects. There are interferences in any analytical method you have to be really careful about. But these definitely offer far better selectivity to some of these compounds compared to some of the other techniques. For example, in this living process. So the distinction with respect to whether it's crystal structure, the carbon allotrope, or just different oxidation states comes from the ability of this microvibration spectroscopy technique. But in addition to that, the recoupling is the way we collect the sample, with the right flow rates, and being able to concentrate that sample into a smaller area that allows you to put the entire sample, and then convert that to the air concentration that comes from just being able to sample the way, couple closely with the spectroscopy. So you're able to do the air concentration measurement as well as a selective identification. I hope I answered that question.
Okay, we have a question from Zoom, so I'll read that first. Many thanks to all the speakers for the wonderful presentation. My question is for Dr. Navesh. Is there a good tool to concentrate or detect airborne pollen in real time?
In real time, it's possible. Japanese company did develop a sensor, and we are also working on developing such a sensor, in which you need multiple wavelengths to detect this distinguish between pollens and others. So this work is in progress, but it is already a commercially available sensor from a Japanese company. If you're interested in sending an email, like I send the link and stuff, like yes. But if you cannot be 100% sure about the pollen type and the distribution, but certainly you will know the concentration of pollens present in there.
Thank you all for great informative talks. I have two questions. So first question for Dr. Kumar. I very well agree that when you want to look at the health effects, it's not just one organ which you would want to look at. It's multiple organs. So when you showed us few data about differences in respiratory health issues, when you were comparing to child. So I would want to ask, let's say if your particle composition changes, let's say if I'm exposed to diesel exhaust as compared to particles just from a dust event, how would your breathing pattern change? So if you have any.
Yes. This is an excellent question, and I give an example. Let me first focus on the eyes, and then I can elaborate on the asthma. When I was telling you earlier that somebody is complaining he has severe dry eye symptoms in Baltimore versus Miami, the mechanisms are very different. In case of Baltimore in winter time, you have very low humidity indoor and spending most of your time. So it's the excess peer evaporation that's causing your dry eye. On the other hand, in Miami, when you're high in moisture and high in microbes, fungi mold floating around, when you're exposed to this, that also triggered inflammation resulting into dry eye. So to answer your question about the asthma, yes, the mechanisms will be different. As I was telling you earlier, all particles are not the same. For example, if you're exposed to diesel particles, your child is exposed to diesel particles and have asthma, likely within 30 minutes to a few hours, he or she will be landing up in ER unless you have some controlled therapies. In case of dust, for example, you go to say on the beaches, those particles are unlikely to trigger a child's asthma because they are salt-rich particles. They do keep your airways moist, and so particle mass is not the same, and the severity of the impact will not be the same because simple dust, a vast majority of them will be trapped in cilia in your and nasal nostrils. Diesel particles, most of them will be smaller than 2.5 microns. So how much of those is delivered is different depending the source as well as the type.
Okay, thank you. I have another question with Dr. Euclid Fernandez. So you mentioned about biocascading factor and David. So in David, you said that we can do in vitro toxicology analysis, but is Handix aerosol devices working forward for site resolved in vitro toxicology analysis? Because if we used Vivaz, eventually the final particle coming onto the cell would be like it would not have the same size as what it was entering. So your size has changed. But if I am interested more in what size of the particle is impacting what kind of cell.
The particle size is not gonna change from when it goes in to when it comes out. So you will still have, if you dry the water on the particle, your particles is still 100 nanometers. You want to go up. So when the particle hits the circle and that water dissolves, you end up with the same size of the particle. So that water is only to make it easy for us to impact it, but the size of the particle that's going to expose the cells, it's always going to be the same. It's going to be the original size of the particle. So if you have a flow with 50 nanometers, 100 nanometers, and 1 micron particles, all those 3 sizes will be the same when they deposit in the cell once the water dissolves. Now, the biocascade is something we are developing right now, so it's still not commercially available, but hopefully we will work with Handix and it will be so as accessible to the device of the cell collector. The cell collector is a different system, but still we're working with Handix with C1. So hopefully that will be also commercially available in a nearby future. So.
I have a question for you, Lorenzo. Couple of questions. So one of the ALI systems you had, is that designed for ambient air?
No, you can use it right now. We'll be testing it in the lab. So right now we'll be testing it in the lab, but it's basically you can take it outside. The only thing you will need to is have, once you finish your exposure, they're using like portable and you're using portable incubators. That once you take yourself with you to the site, you expose it on your system, whatever you want, them in a room, outdoors, and then you put it on its portable incubator and then you put it to the lab. So you can say that system is basically more or less the size of the prototype Vivaz that I saw. So it's something you can take it anywhere, wherever you want.
So how would you, let's say, let's say if you use that as a ambient condition, how would you separate the sort of the effects of gas-based feces from the aerosol. You're gonna have to use it in the urban front to.
Yeah, well, we haven't done that much with study of the gases right now. You could, we did a couple of tests with some activating charcoal in urine ahead of it, if you want to really remove it, but remember, basically, maybe it will be more effect of on the ally system because we are getting the flow, but the flow barely touches the surface of the cells. Basically, the particles hit it because they are going through fast, so they overcome the flow changing towards the X. So we,
Probably the residence time is small.
the resident times, yes, it would be great to do some more tests with to see the. I mean, when we have used anybody in the lab, they use HEPA filters and some of our control. When we say free air means that it's a HEPA filter, still vapors coming in. We have not seen under our control environment. We haven't seen that effect of the vapors, but yeah, it would be good to do that.
I don't know. On that. So when you calculate the dose, micrograms per square centimeter, is that sort of the nominal dose, like you take the actual mass flux and then the area of the cell and. So is it like, I mean, this is probably true for all ALI systems, the actual dose that the cells are exposed to. How do you calculate that? I mean, you could have a high nominal dose, but you could have just the way, you know, maybe orders of magnitude lower actual dose in which the particles are interacting with the cells.
Well, the ally, the surface to the cell. On the other system, the surface, the position surface of the cell.
What's the cell density?
I don't know, depending what quality, what cell culture and so on. So you will have coated surface to the cell.
Yeah.
I don't know, 10 million, 5, I don't know what the bigger number.
It's a continuous form.
It's a continuous form surface that is continuous, it's a continuous coating of the cells. So all the particles are deposited on the surface of those cells. If you have a complement.
Well, you have to see the confluence. If it's 100%, your cells will begin to die.
Yeah.
You have to have roughly 75%
Exactly.
cell confluence before you begin to expose them. But there's some tricky parts too that we can discuss offline.
Yeah, so the people are doing that. Basically, you are depositing everything on the source. So that's kind of how you start this study goes for 4 centimeters. Now, it would be good to do later on some studies that we did usually in Kansas systems some little microscopy to see how many particles fall into different cell parts, you know, like the mitochondria or how many are getting on the macrophages, but that would be something that it would need a little bit more work on the toxicology part to see the real particles that go through the cell membranes into the internal parts.
Yeah, and then you need 3D imaging for that kind of thing.
We do not see.
Okay, I have the privilege to ask the last question and it will be quick because we have different sectors from government, industry and academic. Can I ask everyone, you know, very briefly, 30 seconds, what do you think the other sector can help you, working with you to advance better health for the public? I'll start from
So for the better health of the public, all we are discussing, if there is no health implication, we will be talking about it. So ultimate thing is the science, the data we generate can we really use it in real time and inform people and other stakeholders to take decisions, ranging from doctors, say, if I am becoming a patient today in the clinic, knowing what's outdoor or indoor in this child's home, not only I will treat and diagnose the disease, rather I will be also managing and engaging my patient in preventive strategies. So in other words, how to effectively translate our science and data into public health benefits. So that's a role we can play, industry, government, and everybody else.
I think that what I think that we can collaborate is we need to know what is needed to bring awareness to the people. Like, you know that there's some health related problems with collecting PM and toxicology, but there are no tools for you to give you, enough data. So what would you like the AI system to do to yourself? oh, I have a filter, so I'm a filter required in this time, this thing, and this thing, and
Right.
too long, and I need for a queue response in like 5 minutes, this you have. So you come to me and I will try through the getting money from the government, usually, to build something that will meet your needs. So I need the people in academia to come to me, or academia all over, with what you need us to give the tools for you to go for what.
Yeah, I guess, I sort of echo those comments. I mean, I think, from my standpoint, my wish list is probably more big, and I have shared a little bit on the last slides, but I think what's really needed is, and we are doing some of that, just maybe slightly in different context, some of the stuff that you are developing and others are developing these new techniques and methods that, in a number of contexts are providing much better measurements, but in sort of the workplace type of occupational settings, we need more of that, especially focusing on cost-effective solutions that allow more population studies. It's a very different paradigm that, it's not, and maybe you could claim that there's a lot of funding to do that. But that's what happens. I mean, it's a chicken and egg problem. But often that's broken when you have disruptive technology, you have something that's, much better than that you've done by an order of magnitude. I mean, one of the things that I forgot to mention, one of the instruments, the Spark instrument, it's there is a lot of potential there to reduce the cost. You could pull that whole $1,000–$2,000. And when you sort of suppose that with the existing paradigms, it really allows you to do much better measurements, but then that industry has to do that. Someone has to take it from where we left off to further do the engineering and make mass produced instruments that allows us to do these large data populations. So you got me on my soapbox.
Can I add one last point? Yeah, with regard to, for example, the data we collect in people's home, indoor and outdoor, we provide them a report. And then based on that, we give recommendation, hey, we are finding these kind of things and these small measures, 3-4 different measures. There's a website, it's called dry.miami.edu, security management button. This is where they see the information, plus they also see the report, what we have found in the home into the gases, particles, silent type distribution. So in other words, that helps them guide some, take some of the measures to reduce their indoor air pollution and improve the economic quality.
Okay, that's all we have for this session. Please join me in thanking the speakers and planning survey. And we are going to take a short break. We will be back at 11:10 for the tutorials. The students ready to stay, and hopefully some of the senior can also stay to help us with the tutorials. Let's take a short break. Okay, everyone, we're going to start the tutorial.
Yeah, so we can continue the discussion afterwards. This is mainly dedicated for graduate students using our master's students or undergrads here. So yeah, so I will cover some fundamentals of the aerosol sighting methods, and then Dr. C.Y. Wu is going to give the tutorial on the condensation measurement of the aerosols. And so we're trying to separate the jobs in this tutorial. Nice to see everyone joining the workshop. So my name is Yang Wang. I'm a system professor in the Technical Environmental Materials Engineering here at the University of Miami. So in my tutorial, I'll just briefly cover the aerosol sighting. Well, focus on the sighting side. So first, in terms of references, so here are 2 books that I listed here. So, I found it very helpful when I go through these instruments or go through these slides here. So the first one, aerosol measurement principles, techniques, and applications. Actually, this is edited by Pramod Kulkarni. So he's also our invited speaker this morning, and also Paul Barron and Klaus Willicke. So I think think this is a book that nicely covers all different types of aerosol instrumentation. All right, so the second one is atmospheric chemistry and physics. I think for those who are working with ambient aerosols, you probably already have a copy in your office. And this is written by Stanfield and Pattis. Also want to acknowledge Chen Wang at Washington University in St. Louis. So quite many of my slides are actually coming from this class of the advanced aerosol instrumentation. Okay. So first, just following from what Dr. Pratim Biswas introduced yesterday, right? So one of the most important parameters that we're interested in terms of aerosol characterization is their size distribution. We want to know what is their size and also what's the concentration in different sizes. And there is a series of processes that can lead to different size distribution. For example, we have the nucleation mode that is mainly coming from gas to particle conversion, right? We're very interested in new particle formation, especially in the atmosphere, because new particle formation contribute to more than half of the condensation nuclei. So it has a pretty significant implication on our planet. And then these nucleation mode particles can further grow through condensation, coagulation to apical mode aerosols, and then through the cloud processing, right? So these apical mode aerosols can get converted into accumulation mode, and then in the larger mode, where of course, we can have this weak mode donuts or alcohol emission seed spray aerosols. So yesterday we also discussed it might be quite challenging to monitor these larger particles too. So in order to understand all of these processes and further understand the health and the climate impact of aerosols, we have to be able to measure the size distribution very accurately, right? But how do we measure them? So it turns out that there are quite many methods to quantify the aerosol size distributions. So the commonly used ones include the optical method, right? So you can use the scattering absorption method to find out what's the size distribution. You can also collect these particles offline and then use SEM, TEM to take pictures of the particles, get their morphology and sizes. So you can use aerodynamic method, right? So this is going to rely on the inertia of the particles. And since it's using inertia, so we do not really get to very small size with this method. And then it's going to be the electrical method. And I think one of the most commonly used method in electrical method is going to be the electrical mobility classification. So due to the time limit, I'll just cover the third and fourth method, the aerodynamic method and the electrical method. So first on the aerodynamic method. So since we're using inertia to measure the particle size. So yesterday, Dr. Biswas gave some introduction on the drag force, right? So basically, if you introduce a moving particle into a stagnant environment, then because of the drag force, so it's going to try to stop the particles. And the particle can travel for a short distance, right? You can stop it even analytically to find out how long it takes for the particle to stop, right? So basically, it's a function of this characteristic time, the initial velocity of the particles, and the time that have gone through this process, right? So the SP here is the stopping distance. So the characteristic time is further a function of particle size, the density, so basically related to the inertia of the particles. So the larger the particle, or the heavier or the more dense the particle, then it's going to have a larger characteristic time. So their stopping distance is going to be longer. So we can use this method to classify particles, to basically collect particles based on their size. So one simple example is that we can put a plate over here, and then we can introduce a flow or jet contain some particles. And then the heavier particles, larger particles, they will get collected because their stopping distance is longer. So they cannot really make this bend here to bypass through this plate. And then basically the plate is going to act as a size cutter. You can set it to be, for example, 10 micrometers. So you can collect particles larger than 10 micrometers. Right? So this is a single stage impactor. So what we can do is we can combine a series of impactors. For example, the first stage can collect particles of around, let's say, 20 micrometer, right? And then the smaller particles can go through, and then you can notice the difference here. So the second stage, you're going to have a narrower nozzle, which basically accelerates the flow. The particles are going to have a larger, higher velocity, because that's going to boost up the inertia, right? And at the same time, the particles over the stage here, the distance between the inlet and the stage is shorter compared to the first stage. So you can collect further smaller particles. And then even smaller particles can go through the second stage, go to the third, and then to the final stage. Okay, so basically the final stage is going to have a very narrow nozzle and then also a very short distance between the inlet of the nozzle and the outlet of the nozzle and also the plate. So based on this mechanism, so each impactor can have their own cutoff sizes, right? You can have multiple stages. I think the TSI currently have the Moody impactor, right? So they have the 10 stage or 13 stage, right? So 13 stages, final stages are designed for very small particles, below 50 nanometers, right? So the first stage can right now is set up at 18 micrometers, right? So in this way, after some type of collection, you can measure the mass that's collected on each stage. So right now, also TSI has this QCM-based Moody impactor. So you can measure the mass based on the real-time porous crystal microbalance. So you don't have to take off the stage and do the measurement anymore, right? But there are some concerns. And one major concern is the particle constant, because when you're measuring solid particles, right, they're very rigid, they can bounce around. So you can't really collect these particles. And then they can get like onto the wall. And then if you have some aerosols that are concentrated in certain sites, let's say 5 microns, then you're going to get a lot of mass here. It might overload this stage here, right? So, and the other issue, I would say issue here, but the property of this cascading factor is that when you go to the very small side, then you're going to have a very, very narrow nozzle. Particle speed is going to be very high there. But there's going to be a limit for the particle speed, because the high speed or the high flow is just going to be the choke flow when the velocity is just speed of sound. But you can't make this speed even higher. So under that situation, or even before we reach that speed of sound situation, you already have the compressibility of the air, right? So the drag force or the derivation of these equations are going to be different. So on this topic, Dr. Pratin, this was PhD dissertation actually trying to analyze the high-speed cascade packet system. And download this dissertation and take a look at how the equations are derived. So I think here, this table is listing the different geometries or the speed or the like the Reynolds number for different stages of the Moody impactor. That can go from the first stage where we have 10 micrometers, right? That's the first plate to collect particles, and then the final stage that's 56 nanometers. Right? So you can have this nice table to trace what type of or what's the size of the particles that you measure, and then you can relate the mass that you collect in different stages and get a size distribution. Okay. So as we said, the cascading factor might have some issue when the particles are bouncing, right? So one way to solve that or to alleviate this issue is to, instead of measure the mass, we give some charges to the particle. We're still using these impactors, but now we give charges to these particles through a Coulomb discharger, right? So you can generate unipolar charge, several unipolar charge on the particles, and then there's a charge distribution. For example, larger particles carry more charges, smaller particles carry fewer charges, right? And then at each stage, we have the electrometer to collect the current. The current is related to the concentration of the particles, and then through these electrometers, you can get, or through these currents, you can relate to the concentration of the particles, and then get the size distributions. So recently, there's a new instrument, I would say relatively new instrument called the aerodynamic aerosol classifier. This is commercialized by combustion. So this instrument also uses the inertia, although it looks very similar to the CPMA that we have in their company. But this one actually classifies particles just based on the inertia or the aerodynamic process, aerodynamic mechanism. So what happens is that they have a rotating drum here, right? So you can think of this as a washing machine. Right? You're spinning the water outside because water has higher inertia, but you're not necessarily spinning the air molecules outside. Right? Smaller particles, they will stay there. So particles are introduced from here, right, from the inlet and then to the, let's say, the outlet or outlet at the inner cylinder. And then because of this spinning or this rotation of this instrument, larger particles will get collected onto the outlet stage. Smaller particles, they can follow the streamline pretty well. And then they have an outlet here at the outer cylinder. So basically, only particles with certain inertia or aerodynamic sizes can get exited from the outside here, and then you can use the particle counter to measure the concentration of that particles with specific aerodynamic size. Right? So this is another plot of the trajectory of the particles going through this AAC here, right? But basically, for conceptual, this is still using the inertia. So that's why for this instrument, the lowest height we can get to is around 25 nanometers. And you can imagine under 25 nanometers second, the spinning speed is going to be very high. Right? So that's the limitation for using this instrument to look at very small particles. So the APS, I think most of you saw that at the TSI booth, right, yesterday. So although APS is using the laser to detect particles, but they're not necessarily using optical mechanics to detect them. So essentially, they're still using the aerodynamic method. So they have two parallel laser here, and then you have particles getting introduced through the nozzle and they're running high speed, around 150 meter per second. And then you have these lasers here also dive to measure what's the time the particles go through these two lasers or go through this distance. So basically you can measure what's the time that's needed to go through these two lasers, and the time that's needed is further related to the inertia of particles. And this is, for example, the left-hand side, the acceleration, right-hand side is the acceleration based on the force derivation, right? You can accurately calculate out how does the time correspond to the size or the aerodynamic size of the particles. But there's one thing that we should pay attention is that the APS is designed for solid particles. So they can have almost 100% pumping efficiency of 100 for solid particles. But when you're going to the liquid droplets, then because of the high speed here, some liquid is going to break up, right? So you can see the pumping efficiency is going to decrease, and then the nozzle loss of these liquid particles are going to increase. So we should pay attention when we're measuring liquid droplets, right? So this breakup of the droplet is related to the Weber number, which is further a function of the speed of the particle, density, size of the particle, and also the surface tension. So here, basically, these are the instruments that we use for the aerodynamic method to size them. So you can see the limitation is that we can't really go to very small sizes because their inertia is too small. So that's why we use these electrical methods to look at the even smaller particles. I think right now, the gold standard for people to measure the aerosol sizes, especially submicron aerosol sizes, is the scanning mobility particle sizes. The TSI also showed the SMPS yesterday. So basically, the SMPS is composed of these three major components. You have the DMA, the differential mobility analyzer, that classify particles based on their mobility size. And then you have the condensation particle counter, which is using the vapor condensation to grow the particles. CY is going to cover that later on. You also have a classifier, which provides the high voltage flow through the EMA. And then here, this is a schematic diagram of this EMA that's shown here. This is also a lone EMA. You have two concentric cylinders. The inner cylinder is connected to a certain voltage, normally high voltage. The outer cylinder is ground. So this way, touch the outer cylinder will get shocked. So then the aerosols are being introduced through the inlet upstream of this EMA here. So here I'm zooming in to look at just one side, the left side of this EMA here. So this boundary here, this is the outer boundary of the inner cylinder, right? And then this boundary here is the, I would say the inner boundary of the outer cylinder. So the aerosols are getting introduced here into the classification zone. If you look at the process of the particles getting classified, so you can have ambient aerosols or aerosol coming from reactors, and then they can first get charged. We can use radioactive materials, for example, krypton, polonium, and then yesterday, TSI showed the charger that's using the soft X-ray. So these are the chargers that can give a steady state charge distribution of the aerosols. And normally we assume that most of the particles carry one charge, and most of the particles are neutral. So for the charged particles, we can assume that they carry one charge on, can be both positive or negative. And then after these charged particles are getting introduced into the system, if you think about the neutral ones, they'll just go along the edge here, so they won't have any effect because they're not affected by the electrostatic force. But if you have particles of different sizes, then smaller particles are going to move faster towards the electrode because of their higher electrical mobility. Larger particles are going to move towards the center electrode at a slower speed, because of their larger electrical mobility. There's one thing I forgot, that for the DMA here, you also have a clean sheath flow that's being introduced from the upstream of the DMA. So basically, these particles are going to have vertical velocities that's induced by this sheath flow. Okay, and normally the sheath flow is much higher compared to the aerosol flow. So because these particles are generally very small in the sub-micron range, so their vertical velocity are going to be the same. So there's no difference in terms of their sizes. But the smaller particles, as we said, if they're having the same electrostatic force applying onto particles with different sizes, then the smaller particles are going to move faster, right? So under a certain voltage, you can imagine only particles with certain sizes can get classified and exit through this access slit here. And then we can introduce these smaller dispersed particles into a condensation particle counter to count their exact concentration. And then if you scan the voltage, if you change the voltage, then that's going to correspond to a particle of different size. If you scan the entire voltage from 10 volt to 10,000 volt, then you're going to get the size distribution of the particles. Okay, so we mentioned about electrical mobility quite a lot, but what is electrical mobility? So the electrical mobility is defined as the migration velocity of the particle normalized by the electric field strength. Because if you imagine a particle that's moving in the electric field, then if you have a higher electric field strength, then they're going to move faster. But we want to exclude the effect of field strength. So that's why we normalize that by the field strength. So this is going to be some property that's just intrinsic to the particle themselves. So this is how we define the electrical mobility, right? And then if you consider the particle movement in the classification of the DMA or under steady state, then this electrostatic force is going to be balanced by the drag force. And then by assuming that particles are in the Stokes regime, so these two are going to be equal to each other, and then you can calculate what is this migration velocity. And then based on this equation, and further normalize that by the field strengths, and you can get this exact form of the electrical mobility. So mobility is going to be a function of particle size, not only in here, but also in this C term. So this is the quantum slip correction factor, which is further a function of the Newton number, and then a function of the mean free path and particle size. Particle size exists both in the quantum slip correction factor and the particle size. But overall, there is going to be a one-to-one correspondence between particle size and mobility. So if you have larger particles, then they have smaller mobility. If you have small particles, they have large mobilities, right? And then in terms of their size, you can also divide them into the continuum regime, where the particle size is very large compared to the mean free path, or they're in the premolecular regime, where the size is comparable or smaller than the mean free path. Right, so in different regimes, then you're going to have different relationships between the mobility and particle size. But overall, there is going to be a one-to-one correspondence. Whenever you know mobility, you know particle size. Okay. So here I'm showing you diagram of the DNA again, right? So now we can try to understand why smaller particles faster. So if you consider we're first introducing if we have smaller particles, right? So then because of the balance between the electrostatic force and the drag force, so no matter which side we're talking about, if we assume that they carry one charge only, then their electrostatic force are going to be the same. Okay, so this is not dependent on side. But on the drag force side, this is functional side. So if you have larger particles, then they have a smaller vibration velocity, which means that they're not going to move much towards the horizontal directions. They're just going to follow or stay close to the outer electrode. But if you have small particles, they have a very large migration velocity, which means that they're going to move towards the inner electrode faster. So in this way, you can separate particles of different sizes and then get them collected, exit, and then you can count the particle concentration. So currently the TSI SMPS have two different types of EMAs. So you have the low EMA, which is targeted for particles between 10 to 600 nanometers, or the nano EMA, which is targeted for particles between 3 to 200 nanometers. So I see these two EMAs, or I feel that the difference is just the length of the classification zone, because if you measure very small particles, then you're going to have diffusion of these particles. So to reduce diffusion, you have to make the classification zone shorter, so they stay for a shorter time in this region here. So you have smaller diffusion happening. Okay, so here I introduced this cylindrical EMA, which you have to make sure that the distance or the gap between the inner cylinder and outer cylinder should be the same, right? And sometimes in terms of machining, this might be quite challenging. So that's why there are other designs of the EMA. There can be parallel plate EMAs. I'll introduce that at the end of my talk. And then there's also this radial EMA, which is invented or also worked by Caltech and Aerosol Dynamics Incorporated. I have a few colleagues here. So basically for the radial EMA, instead of introducing a sheath flow from up to down, you have the sheath flow that's introduced from the side, and then they will get exit from the center. Right? So the particles are introduced from the bottom plate here. This is the inlet, so they are circular. And then the particles are going to move inward because of the electrostatic force, and also the sheath flow, so they can get collected. So the advantage for the radial EMA is that you don't need this large geometry of the EMA, so they're more compact and potentially lower cost. So in terms of the operation of the EMA, one pretty important factor that we need to consider is the transfer function. Actually, transfer function applies to all different instruments. Right? It's just the DMA let's say, if you're trying to make your own SMPS system, you have to consider the transfer function of your EMA, of your operating condition. So the transfer function is defined as the probability that a particle of mobility ZP that will be transmitted from the aerosol inlet to the sample monodisperse outlet. Okay, so the easier way to rephrase it is that let's say the probability of a particle of, let's say, 100 nanometer particles that can get transmitted through this classification zone when you are scanning the voltage applied onto the EMA. This is, although we're talking using about particle size, but particle size have a one-to-one correspondence with mobility, right? So you can see that you can imagine, let's say if the EMA is set up to classify particles of 10 nanometers, then it's very unlikely for 100 nanometers to go through, right? So the transfer function will be 0. But if it's set at 100 nanometers, then almost all of these 100 nanometer particles, so the transfer function will be close to 1. But what about 99 nanometers? What about we set up the voltage at 99 or 101? What is the transfer function? It's not going to be 0 because there is the distribution of the aerosol flow here, right? And it's not 1 either because it's not set up to collect 100 So an easier way to demonstrate this is that, let's say we set up the DMA to classify 100 nanometer. In this dash line is the axis of symmetry here. So this is the inner electrode, this is the outer electrode. Let's say we introduce 100 nanometer particles from the inlet, right? They will travel through the classification zone, and the DMA is set up to collect 100 nanometer particles. Under ideal situation, all of these particles will get affected. But now, if you think about the trajectory of 99 nanometer particles, right? So they will move faster towards the central electrode, right? So some of them will get collected by the outlet, right? Which means that when the DMA is set up to collect 100 nanometer particles, there's going to be particles of different sizes. And then some of the larger particles, 101 nanometer particles, some of them will get collected too. So because of that, there's a nice derivation of the DMA transfer function. This is Newson and Whitby. I feel that this is kind of like a Bible paper for DMA operation. They elegantly show how to derive the transfer function by just using the conservation of the streamline, right? So I encourage all the students to read this one and try to do some equation derivations over there. So they found out that when the aerosol inlet flow rate balances the aerosol outlet flow rate, then the transfer function of the DMA is going to be triangular, which means that if it's set up to collect 100 nanometer particles, then you get 100% collection efficiency. By the same time, you can collect particles, let's say, as small as 95, as large as 105. So the width or the distribution of the resolution of this peak really depends on what sheath flow rate you're talking about, the sheath flow and aerosol flow. So ideally, we want this transfer function to be infinitely narrow. We want it to be accurate, no particles of other sizes to get classified. But we cannot really do that. Okay, so the resolution is just a ratio between the sheath flow and the aerosol. For example, if you're taking 1 liter per minute by the CPC and use a sheath flow of the given 10 liter per minute, then the resolution is going to be 10. And so this is how the transfer function is going to look like. And then you may wonder, why is that important? Why can't we just use the whatever the CPC tells us to report the size distribution? So an example can be, let's say now we set up the DMA to 100 nanometers. So I used to use the older version or earlier version of SMPS. You can use a dial to set up the particle size, whatever you want. I think the newer version now we have the DSI has a touch screen. Still prefer this one, right? So you can directly get control of this process here. So let's say we set up this DMA to collect 100 nanometer particles, and then the CPC downstream will give you 100 particles per centimeter cube. What does this mean? Right? Does it mean that the size distribution function is 100 particles per centimeter cube? No, because the CPC is collecting particles of different sizes. So it doesn't mean that there are 100 particles for this specific size because of the transfer function. And also, because of the transfer function, you're going to have particles of different sizes. And at the same time, you need to consider the charging efficiency of the particle. Not all of the particles carry uniform charge. There is also particle loss in the system. Transfer function, we need to consider that, and also the counting efficiency of the species. So all of these are convoluted, right? And so also we need to consider the scenario that when we classify very small particles are going to diffuse. And then let's say we set it up to classify 3 nanometer particles, and then they are going to move around. They have a random motion. So the peak of this transfer function is not going to be 100% anymore, because they just get random walked, right? And then get collected either upstream or they can go downstream. So because of this diffusion of the Brownian diffusion of the particles, then you're going to have what we call the broadening of the transfer function. So this is a result that's coming from Rick Flagen, right? So basically, if you have the perfect triangular transfer function, then they're going to look like this. But if you're classifying smaller particles, let's say smaller particles correspond to lower voltages, then you're going to have broader than very broad transfer function. Okay, so we need to consider this when we try to calculate the actual size distribution of the particles. And here I'm showing you the charge distribution, because not all of the particles carry charges. Right? Some of them will carry +1, -1, +2, -2. Right? So what we classify downstream of the TMA will be particles carrying unipolar charge, bipolar charge. So there has to be some charge correction going on here. And because of that, now if you imagine what you measure at the CPC under 100 nanometer setting. So this is going to be the number of particles detected by CPC. So as we said, it's definitely a function of particle size distribution. Okay? It's related to the size distribution. But we need to consider the penetration efficiency, because particles get lost in the sampling line. We need to consider the counting efficiency of the CPC. We need to consider the bipolar charging probability of the particles. Right, so some will carry 1 charge, some will carry 2 charge, 3 charge, all the way. I mean, it can be many charges. And then for each charge, then there is going to be a transfer function of the DNA, and we need to consider the broadening of the transfer function. Okay, so this is going to be a convoluted integral of all of these parameters. What we know is what we count from the CPC. What we don't know is the size distribution. But for the others, penetration efficiency, counting efficiency, we can calculate them. So basically, it's converted to a problem of, you know, the integral, but how do you find out one parameter inside this integral? Okay. So what we can do is we can separate this integration to different size bits. Right? So you can set up the resolution of your measurements. And then further, we assume that the size distribution function is a constant within this narrow segment. Because a lot of times when we measure particles, it's going to have a rather broad size distribution. Okay, so we convert this integral into a summation of all the, let's say, particles, all the different sizes, and then we take this size distribution function out to here. And then this is going to be the size distribution function in that specific size thing. And then you're going to have an integral here. But now for these parameters, we already know that we can calculate that in some integrations. So basically, this is the CPC value you read at 100 nanometer, right? And then you can do 90, 110, so you can scan the DNA, scan the voltage to get a series of concentration. And then you can calculate, you can try to calculate what is the size distribution function based on what you measure from here. So then this problem is getting converted into a matrix calculation, because this is what you measure at different EMA voltages. You want to solve what is the size distribution at different sizes. And here this matrix is related to all the functions that we talked about, the transfer function, and then the charging probability, penetration, and counting efficiency. So in terms of how to solve this matrix problem, there are several approaches. So the widely used one is a 2-mean inversion method. And then I think there are also studies using these non-negative least squares matrix inversion. So right now, I don't know what TSI is using in terms of the inversion matrix. I saw some description in the manual, right? So there are some assumptions being made over there. But what I want to say is that whenever we use SMPS and the size distribution, this is what we get. But there's actually a complex process that we go through this data inversion process. And if you want to make your own SMPS, a lot of groups there, they buy the separate EMA parts, they have a CPC, and then you can scan the voltage. Then you have to consider all of these factors here and create your own inversion methods, right? So I will use the rest of them. How much time I have here? 3 minutes to just cover some other methods to measure size distribution. So During my postdoc, I got to work with Jian Wang to basically work on an instrument that can do fast scans of the size distribution. So again, if you look at the size SMPS, to measure the size distribution, you have to do the scanning, right? So now the newest version of the SMPS can do 30-second step, which is already very quick. But most of the time, people still use 1 minute. So for this time resolution, it is acceptable for ground-based measurements, right? We don't care what's going on by doing yearly based measurements. The thing is that if you're trying to measure aerosols with transient properties, then you may have a problem because, for example, if we're doing biomass combustion just last, some of them last for a few minutes, right? You can probably just get a few samples. And also for the aircraft-based measurements, since the aircraft move with a very fast speed, right? So if you still use 1 minute scan, then the aircraft's already 6 kilometers away. And you might go through a lot of clouds over there, and then you can't do the entire size distribution. So we're trying to work on an instrument that can do fast scans to achieve the 1 Hz resolution. And also we want to measure the submicron range, especially below 100 nanometers. So there are instruments that can do this. For example, you can replace the CPC by these electrometers and then just plot the particles based on different mobilities. The thing is that the electrometers have limited sensitivity. So you can't really get to very low concentration, especially in our ambient environment. We don't have a lot of particles to go to clean environments to do the sampling. Then these electrometers probably will require at least maybe 1,000 particles at each specific or in each specific band phase. So that's why we still want to go back to the condensation method, which is CY will cover later on. So I got to work with this fast integrated mobility spectrometer. So again, this first prototype is made by Pramod, and big shout out to him. So I think this is somewhat a game changer in the aircraft sampling of the aerosol size distributions. So here, instead of using the cylindrical DMAs, use parallel plate DMAs. And then because of the high voltage, the particles can then again get separated to different sizes. And then at the same time, we have this sheet flow, particle sheet flow that's going down. The sheet flow is saturated with the ethanol, alcohol vapors. So what happens is that then after these particles are separated, they go to a condenser, and particles still move on, but there's no voltage anymore. So they just go through this horizontal, vertical movement. But because of the lower temperature, so the vapor can grow onto these particles and make them supermicron droplets. At the end of the parallel plate, we have this horizontal laser to shine across this gap between these two cylinders. So basically, we're going to see a bunch of particles get included in this image here. All right, so this is the schematic diagram of the flames. I think this animation is probably made by Pramod. I got it from here. And then basically introduce these particles to the charger, they pick up charges and then get separated inside the classification zone, and in the condenser they grow to supermicron droplets. So the CCD camera, a very fast image of these particles. So the location of the particles related to the mobility, because that's how we separate them. And then mobility, as we said, there's a one-to-one correspondence between the particle size and mobility. So you can get the particle size on the image. Right? So now if you're introducing particles of exactly 100 nanometer particles or exactly 100 nanometer, now you can see all of them are going to align and near a very narrow region. Okay. So basically from the images, you can get the size. So if you do ambient measurements, you're going to see a bunch of particles getting distributed here. And then from the images, you can back up or go through this data inversion to get a size distribution. So this instrument has very good agreement with the SMPS, except that we do these measurements every 1 second. We don't need to do the scan. And then we also put this instrument into the aircraft, onto the aircraft, and did a few campaigns. So again, these instruments can give you very, very high time resolution size distribution and integrated size distribution naturally well with nutrition by the cities. So this is a closure of the different instruments. Another example I want to give is about the diffusion of the particles. When you're classifying particles based on measured mobility, small particles are going to diffuse around, right? So we said very small particles, then their diffusion transfer function is going to be very broad. And that might cause some accuracy problems. So the easy way to solve that is to reduce the particle residence time. So basically, we just make this region much shorter. And at the same time, we give it a very high sheet flow rate to let the particle travel really fast. So you're suppressing the diffusion of the particles. So for this instrument, so Dr. Juan Fernandez de la Mola at Yale University, so he made this high resolution EMA or high flow EMA. So these EMAs operate at sheet flow of higher than 200 meters per minute. And so for conventional SMPS, I think you can go to up to 30 meters per minute, right? But if you have a very high sheet flow rate and also very high noise, so you can go to very small sizes, down to 1 nanometer. And people have used this to calibrate their condensation particle counter, and Chugai is using this system to calibrate the CTCs, and also we can use that to understand the particle formation. So in our lab, we can use that to understand combustion particle formation. So this is also some new particle formation, but happening in a more engineering method. Right. So with this instrument, we can go down to below 2 nanometers when we introduce particles or when we try to synthesize different type of particles. You can see the very narrow peak of the particles. Right. So we can also collect them by some physical method, take the AFM image, and try to understand the formation pathway. So finally, this summer, so I introduced a few cycling methods and briefly cover the daily inversion. And so there's a lot of equations that you need to derive. And I introduced this faster measurement with higher time resolution. So I probably go over time too much, but I'll hand this to CY to talk about the condensation particle complex.
Okay, everyone, I have a challenge because I'm standing between you and lunch. I'll try to fly through this very quick, but yeah, hopefully we will be able to finish 10 minutes. Let's try that. Okay, so we have heard a lot about the condensation, right? And I want to talk about the principles about condensation. So this is the process of particle size growth due to vapor deposition in the presence of a condensation nuclei or ions. You can see that in your daily life a lot. For example, you can see the plume. That is because the water vapor condensing on the edge particles and making the edge particles bigger, and your eye can see the scatter light from the droplets. And similarly, the cloud formation is shown over here. And when you have a water vapor going up due to the adiabatic expansion and cools down, and then the water vapor condensing on the naturally occurring nuclei and making them bigger, again, more efficient in scattering the light, and therefore you can see the cloud. So this happens all the time, and I wanted to talk about the next one is about the condensation equation. I know this is a lot, but to explain the mechanism, I do have to use a stable equation over here. And I'm going to use the continuum regime as an example. And Dr. Wang talked about different regimes. Here is a continuum regime. We have the second law of fixed second law of diffusion shown over here. And it describes the concentration change, the concentration of vapor change as a function of the time and the location. And I'm going to make us simplify the condition. This is the steady state, and therefore it is the steady state, and therefore this term is 0 over here. And then if you impose the boundary condition at the surface of the droplet, this is the concentration of vapor on the droplet surface, and very far away, this is the infinity, and infinity is concentration very far away. Okay, so that's the concentration difference between these 2 points is the driving force. So solving this equation with the boundary condition, this is the final equation. Okay, this gives you the description how fast the concentration changes over, as a function of distance. Okay. And with that, we can talk about the particle growth, because the particle growth comes from the vapor condensation. So what we have over here is the diffusion, the flux to the surface of the particle or synthetic growth, then the droplet surface, multiplied by the surface area, the surface area term. So I have this rate, which is the number of molecules on particle surface per second. Okay. Then the next one, okay, related to particle size growth. So the volume growth is just the rate we just talked about, multiplied by the volume of each molecule. So that's the total volume condensing onto particles. You can express it as the particle volume. You can also do with the diameter here. And let's take a quick look at the equation over here. This is the partial pressure. So the partial pressure difference or the concentration difference is the driving force. So this is larger, you have a higher rate. And similarly, if you look at the diffusion coefficient over here, and you have a higher coefficient, then you have a higher growth rate. And similarly, you can consider other parameters. But I do want you to pay attention to particle growth by particle diameter and the particle volume. You see, for the particle volume growth, it's proportional to the particle diameter, right? So what does that mean? A larger diameter will grow faster in terms of a particle volume. Okay. So the volume you can have the condensation is higher for larger particles. But if you look at the particle diameter change, it is inversely proportional to particle diameter. So what does that mean? The smaller particle grow faster in terms of diameter. These two are different. These two are different, right? So what is the implication from that? Let's use an example. Okay. So I have a situation. I will start with two particle size diameters, 0.1 and 1 micron. Okay. We want to find out the final diameter after 1 second of water vapor condensation. And this is at the 20 degrees C. And assume the equation we just derived can be used. And there is an unlimited vapor supply for the condensation. Okay. And these are the parameter values. So the particle size growth rate we have from the previous slide. This is the equation. So I can integrate both sides. I get the particle diameter, okay, as a function of time, which is over here. Okay. And now everything here is a constant. I have the diffusion coefficient is a constant, volume of molecule is constant, and the partial pressure difference is constant, temperature is constant in this condition, right? So I have calculated all the terms over here. This is equal to 3.33 times 10 to the minus 5 centimeter squared. Okay, we'll just use this value. Now I wanted to impose it to the students over here. If I start from 0.1 micron or 1 micron, what will be the final answer to the particle diameter? Okay, so you have this term over here, right? And 0.1 micron is equal to 10 to the minus 5 centimeter, right? And you take square 10 to the minus 10. How does it compare to this one over here? It's much smaller. And similarly for the 1 micron, it's also very small. So the answer is based on the numbers over here. If you start from 0.1, you end up with a 57.7 micron. If you start from 1 micron, it also ends with 57.7 micron. Wait a minute, you start from different sizes. You end up with the same size, right? Okay, so I want you to remember this. This is the most important feature of condensation. When you can start from different sizes, but you end up with the same size. You start from different sizes, you end up from the same size. So this is the most important thing about the condensation. So in condensation, the particles grow to the same size, regardless of their initial size. Of course, this is based on the condition that there is sufficient data for the particle size growth. Okay, so if you can walk away from this presentation and remember this, I think you have already done a very important lesson. And now I'm going to do, okay, this is the demonstration, the experimental measurement of the size distribution. Start from a very wide size distribution, allow the particle to grow to vapor condensate. You can see the spread is much smaller, approach amount is first. If you get enough time, there will be one line over here. And Dr. Reza in the morning her slide, it shows that if you allow more longer residence time, slower flow rate, then you will get small, narrower and narrower distribution. Okay, so again, remember this is the key feature of the condensation. So with this, you can apply the two different devices, and this is the CNT or CPC. Okay, you can play with the PSI. This is the principle. You have the aerosol coming in, and you heat this reservoir, so you can saturate the system with vapor. And then later on is the cooler region. You can have the condensation of vapor, and then they grow to the sizes, and you detect it. So this is the principle of CNT. Well, the question is, how do you measure the number of concentration by light scattering intensity? The particle size is different. If they have different sizes, they have different scatter light, right? So how do you really convert that to number of concentration? The answer is you cannot. So how do we use this one? Well, going back to the feature of condensation, all the particle sizes with sufficient vapor condensation, they grow to the same particle size. So they all scatter with the same scatter intensity. You can convert that measured scatter light to the number concentration if you know single particle scattering. Good. Okay. So in that case, can we use the CNT to measure the particle diameter? The answer is no. Okay. What if we combine VM and CNC? Going back to Dr. Wang's slide, you can have the SMPS. Again, if you wanted to learn more, you can talk to TTS. I know it. Hey, now I wanted to impose a question for the audience. If you have an aerosol system and you heat it up and then cool it down, what will happen to it? You heat it up, you have a lot of vapor in the system, right? When you cool it down, it's going to condense. Again, going back to the key feature of condensation, what do you have? Monodispersed. Monodispersed type. So if you want to produce monodispersed type for your experiment, this is one system you can use. Again, vaporization and then condensing to produce monodispersed particles. You can only achieve this using condensation. Okay, now I wanted to switch the focus a little bit different. So we have been talking about the condensation, and the examples I have used is based on alcohol. But can we use water? Well, not really, because the CNT doesn't perform well with water because the mass diffusivity of water vapor is larger than the thermal diffusivity of air. What does that mean? Well, I'm going to use the animation to show that. So for the alcohol, vapor coming over here, you have the condensation of vapor, okay, making it bigger. But if you use water, okay, you have the water vapor, which want to condense onto the wall instead of the particles. So if you wanted to use water for the condensation, using the same device, you cannot do it. And that's why ADA have developed this water-based EDC now is sold at ESI, and they use a different approach. How do they do that? Okay, Dr. Reza in the morning talk about this slightly. So you have the aerosol particles you want to measure. First, you condition it to make it a cold nuclei. And in the next section, you provide this saturated vapor by heating from the wall. You provide the vapor coming from wall, which you can condense onto the seed particle within vapor, and then you can collect them for whatever analysis you want to have. So this is the opposite direction, and that's due to the property of water vapor compared to alcohol. Okay, so alcohol-based CPC and water-based EPC, they have a different configuration because of the principle. But hopefully a student can see this. Okay. And then not just in the ambient environment, in the respiratory system, it happens the same way. Okay. So you have particles coming into your respiratory system, and this is a very moist environment. What will happen to these particles? A lot of them, most of them, it will grow by condensation, right? Then you can deposit it. So again, it's going back to the AES device, the David that Lorinda mentioned earlier. Okay, so you grow the particles and deposit onto the cells, and you can determine the toxicity of the nanoparticles. Or if it's a biological particle, then you can observe the interactions of the cells with the virus or microbes. So based on the principle of condensation. Okay, and there is another cloud droplet device called a digital cell. You introduce the water droplets over here, actually containing your particles, and you make the cloud which has the vortex homogeneous mixing, and then you can produce the homogeneous droplet size due to condensation, and they will settle down. So this is another device that uses condensation. Okay. Right. One last thing I wanted to really point to is it sounds like a condensation is wonderful mechanism, and it is, but there are limitations. Okay. For one thing, it is about the concentration you can play with. If it's too high, then you're not going to get the measurement or the particle size growth you want to have. The other is about the temperature effect. Okay, so the temperature of the surface may be different from that away from the surface. Okay, so here this is the equation. We don't need to remember, it's better to know that the temperature will be different. The previous equation assumed the constant temperature. So incorporating this and then, so this one, if you have a molecule condensing from the gas phase into the liquid phase, you are going to release the latent heat. So what will happen to the droplet with the added energy? It's going to heat up, right? Okay, so this droplet is going to heat up. So what? It's getting hot that the water, that the vapor molecule wanted to condense, has less desire to do so, right? So when you have a condensation, especially when you have a high concentration of molecules condensing onto the liquid, you are going to have a slowdown process because of the latent heat. So if your system is dealing with high concentration particle growth, you may have, let me take it back. If you have condensation in the system with a lot of water vapor, or not necessarily water, the organic vapor also the same. You have a lot of condensation onto the particles are getting hotter and hotter. That's going to slow down the growth process. So you may not get the final size you want to have because of the slowing down. And let's take a look at the difference. Okay, this is droplet temperature minus ambient temperature as a function of a saturation ratio. Okay, if you start from 0 degrees C ambient temperature, okay, or you start with the 40 degrees C temperature, you can see at higher ambient temperature because the molecules contain more energy. And therefore the condensation will heat up the particle more than if you have a lower ambient temperature, right? And also if you have a higher saturation ratio, then you have a higher temperature on the droplet compared to the ambient. So that will slow down the process. Okay, I'm done with this. So hopefully you take home with a few key messages. The first one is condensation produces monodispersed aerosol. And second is the temperature effect. If you have a lot of condensation, it's going to heat up the droplets, and therefore you have to have a slow down process, right? And third thing is water CPC and alcohol-based CPC operate on different designs. Okay. Well, I went over, but it's still within the original schedule time. Hopefully, it's not too long. Any questions? If not, thank you very much. And we are going to have our lunch, and we still have the posters, and the students will be more than happy to introduce their posters to the audience. And we also have the final demonstration for the instrument for the BioSpot and Jen, if you are interested in working with them. Thank you very much. Let's start our lunch.
Okay. All right, I think everyone, let's get started. We're already a little bit over. We're about 6 minutes over already. Sorry about that already. So again. Yeah, so again, welcome back everyone. So this is the last session of the workshop. So again, we're going to kind of be focusing on nanoparticles, nanotechnology, nanoparticle synthesis, and also kind of focusing a little bit, especially in the first two talks, on sustainability and kind of synthesizing sustainable kind of nanoparticles and sustainable formulation. So you'll hear obviously from Orlin, who will introduce, and then we also have speakers from industry, from Eastman Chemicals. So we'll kind of, is this on? Yeah. So we'll kind of hear from both of them. So I'd like to first introduce our first speaker, who's the keynote for this session, is Professor Orlin Belev. Orlin is a Frank and Doris Culverson Distinguished Professor at NC State University. He received his PhD from the University of Sofia, Bulgaria. And then he did a postdoc at the University of Delaware. And then Professor Belev has contributed to more than 235 publications and presented more than 300 invited presentations. His numerous awards include NSF Career, AICHE, Andreas Archivos Award for Professional Progress, and ACS Langmuir Lecturer. And Professor Belev has been elected to an ACS Fellow and MRS Fellow, and his area of research is innovative research in the assembly of colloidal nanostructures with electrical and photonic functionality, biosensors, microfluidics, and nanomanufacturing. So everything encompassing the world of nanosciences, Orlin. So again, it's a great pleasure to welcome Orlin to University of Miami. And again, it's my alma mater as well, so from NC State. So it's a double pleasure for me again. So with that, I'll hand it over to you, Orlin, to talk us through design and manufacture and applications of functional, environmentally benign nanoparticles. Thank you again, Orlin.
Thank you, Samuel, and it's really a pleasure to be here. Thank you. It's really a pleasure to be here. I have never been before to the University of Miami, and I'm very impressed by the campus, the facilities, and the general atmosphere here. So it's really a very good opportunity. And it's not that far away from North Carolina, but somehow the weather was very dramatically different when I was coming. So it's a pleasure to have at least one day of sunshine. What I'm going to discuss is, Samuel mentioned that we have a broad range of research in the CMI group, and I'll mention those. But I'm going to really focus on the environmentally benign, sustainable materials that we try to make and their commercialization. So before anything else, I would like to acknowledge the contribution of the people who have actually done the work, the specific authors of the papers that will be presented here, my research group in general, and the funding. And over the time of our research, we have worked with a couple of startup companies, the latest one being Benanova, which is actually trying to commercialize some of the aspects that I'll be discussing. So I'm going to share our experience, positive and negative, in the regards of sustainable materials. As mentioned here, my research group is not only working on sustainable materials. We have a broad range of interests, generally relating to colloidal science. I'll be discussing different types of particles here, and we do a number of industrially relevant systems. These days also, when it comes to academic research, there is lots of interest in the so-called active and reconfigurable systems, which are shown here. Some of them 3D printed to additive manufacturing, but this is not what I'm going to be speaking about. I'm really going to focus here on sustainable, environmentally benign materials. So this is an aerosol workshop, and our focus here is really not aerosols and more like nanoparticles that are made in liquid. But definitely the type of structures that I'm discussing are coming to aerosol format one way or another, right? Whether positive or negative. And hopefully, even if they come as a negative dispersed type of materials, because they may be used in agriculture, hopefully they are the natural origins of this type of materials are going to make them much more benign than some of the kind of like, almost horror stories that we have heard here with regards to impact of the different types of inorganic aerosols. So I'll be discussing organic particles here. What I'll do is I'll first discuss the rationale that we had when some years ago, we started this topic of environmentally benign nanoparticles. Then how can we scale up the process of making those particles? what would be agricultural applications of similar type of particles. And I'm just going to mention some of the other ongoing research on making particles of special shape by different types of polymers and biopolymers, which we find out to be very perspective. I think I can fix that. Okay. I'm going to be discussing particles which have two natural ingredients, which are very common. And kind of like I would say the mainstay of today's research on sustainability related to materials and nanomaterials. The first of those is lignin. Lignin is, well, I mean, obviously, you can see lots of lignin around because everything, or most of the origin of the brown color that you see around in the soil and in tree, unprocessed tree material and so on, comes from lignin. So lignin is the second most abundant component in natural tree materials and all kinds of plants. It degrades slower and kind of like is released from the from cellulose when it is being degraded or processed, and then you can see it all around. So the things to keep in mind about lignin is it's a very complex material, very undefined chemical composition in the broader sense, and then when it gets processed, it gets chemically functionalized by other means, which have to be taken into account once one start making particles out of lignin. But generally, it's a phenolic type of material. It has lots of carboxylic groups, and the lignin that is being industrially processed, so-called kraft lignin, is going to have sulfide and sulfide groups also present in the material. Lignin is dirt. Well, it is the basis of the dirt around, and it is also dirt cheap. So it looks like a good idea for commercialization. A few years ago, at least, if you try to commercialize lignin and kind of like speak with people, kind of like from the entrepreneurial community about the commercialization of lignin, the joke about lignin was that this is like the joke about how do you make a billionaire. The easiest way, you take a billionaire and make a and then make him or her lose the funds. The idea about lignin was how do you lose from commercialization? Well, you try to make something out of lignin. That time is hopefully over. So this is a very nice presentation of all the present areas where people are trying to commercialize lignin type of materials. This is from an excellent review paper from Orlando Rotos, another connection from NC State University, presently in Canada. What can you use lignin for? I'm going to discuss antibacterial activity here, and then agricultural applications, what is what we have been doing. But there is lots of interest in different types of biomedical applications, which are a little bit trickier, obviously. Also different types of antioxidant and UV protection. So these days, hopefully, the kind of like the idea that you cannot make something useful out of lignin kind of like has been overcome. And I'll show what we try to make out of it. The other type of natural material that is very widely investigated, that we also use in similar type of particles, is chitosan. Chitosan comes from reprocessing of seafood. Basically, that is crustaceans. It is a major component of the shells of crustaceans and also beetles. Now, chitosan, that is not the major component. The major component is chitin. Chitin can be extracted, can be reprocessed, and in order to make it chemically processable, with some functionalization, acetylation, it can become chitosan. So chitosan and lignin, kind of, you can think of this as kind of like, the low-tech and high-tech biopolymer in this field. So lignin is dirt cheap and kind of like very undefined and has relatively poor mechanical properties. Chitosan is kind of like really can be made very nice. It is very strong mechanically. It can be functionalized in very special way. Of course, also the kind of there is difference in the price. Chitosan can be expensive, but it's one of the major components of more high technology type of materials in different biomedical applications, even electronic applications, displays, and sustainable packages, which are also interested, by the way, in my group, but I won't be discussing. So now we take those particles. We want to, well, we don't take the particles, we take the polymer, some kind of form as it comes naturally. That is from the producers. In my group, we're working a lot on the process of what is called anti-solvent precipitation for making the particles. So this is one of the major processes that you kind of like, of course, use very widely in industry. What you do is you take the polymer or biopolymer, dissolve it into a solvent, then shear off the droplets of the biopolymer solution into medium, where the polymer, the biopolymer is going to be precipitated. What is going to happen to trivial result, and this is one of the major methods of making particles or nanoparticles, whichever way you call them, that is kind of like one of the most major industrial way. If you do shearing of the system and different types of hydrodynamic processing of the system during the fabrication, and by the way, that the same may happen in different types of aerosol processes, you're going to have more complex particles than just the simple spherical precipitated material that you have in the general process. If you control a number of parameters which involve concentration of polymer, molecular weight, degree of cross-linking, and so on, you can end up with a variety of interesting type of particulate materials by using a process like that, which we have been describing in other papers that I will not discuss much here, but I'm going to go back to some of the more interesting materials and data might come. Right now, what we're going to do is we're going to discuss the very simple formation of lignin particles by using anti-solvent precipitation. Something that is as easy, once you get kind of the conditions right, as easy to do as anything in the laboratory. And it is illustrated here. So basically, we take lignin solution. We pretty much drip the lignin solution into shear medium, which is going to precipitate it. One of the good things about lignin that also makes the whole sustainability claim here is the following, that the medium for precipitating lignin can be as simple as water. So basically, you can dissolve the lignin in a relatively benign solvent, such as acetone or ethylene glycol, and then you can precipitate it in water. So you do not have volatile or highly toxic solvents in this case. And in some cases, the whole process of making lignin nanoparticles can be completely water-based. You can dissolve it at high pH, as I'm going to mention later on, and then you can precipitate it at low pH. So basically, you have what we call water-water process, where everything that you need to do in order to make particles is water and lignin, and you don't need to use any organic solvent. Even with the use of organic solvent, the whole process looks a bit like that. So this is the lignin solution, in this case in acetone. And then you add water. And our favorite way of kind of immediately visualizing the state of the system, the formation of particles, is using a laser beam, pretty much from a laser pointer. In many cases, and visualizing the scattering from the system. If you do this even with the concentrated lignin solution, you may not see the beam, but the beam here shows the presence of the nanoparticles or particles in this system. So light scattering is, of course, one of our major ways of characterizing the behavior of the system. The size of the particles, as known from the literature and as shown in our papers, the size of the particles in this case is going to be determined by the degree of supersaturation of the system, and basically the concentration of, in this case, lignin, which is going to be precipitated once you shear the droplet. So the higher the concentration of lignin in the shear droplet, the larger the size of the particles. The distribution may change a little bit, but generally increasing the concentration will lead to formation of larger particles, which is one way of controlling the type of particles that you're going to get out of this process. And then here comes the kind of the most important, I would say, concept and idea, which we introduced some time ago, and by now has become quite popular. So you hear about sustainable and environmentally benign materials all the time. At that time, when we started this research, which was around 2010, that was not kind of like such a popular topic. So we believe that our paper was one of the first type of research publications which popularized a similar area. We started this research based on discussions with the EPA, which is also happens to be in our area. And at that time, 2010, EPA was freaking out a little bit about inorganic nanoparticles, and especially about metallic nanoparticles. If you can kind of like hold what has been going on in these years, silver nanoparticle at that time had become very popular as antimicrobial agent. And there were kind of like development of different types of ways of making silver nanoparticles in very simple and rapid way, such as different electrochemical means. So at that time there was even ideas. Samsung was about to start producing washing machines, which are going to make silver nanoparticles during the washing process and disinfect the laundry. Except that what happens after that, those particles have to be carried out with the water that leaves the washing machines and then being disposed in the environment. So there was a big concern about inorganic nanoparticle material reaching the environment and staying there for a long time. So the idea that we proposed and we realized was the following. The effect of silver nanoparticles in antimicrobial sense is done because there is always an oxidative layer on top of the surface of the silver nanoparticles. So you're going to have silver ions being released from the nanoparticles on a continuous basis. Silver nanoparticles, the ones that are most efficient, also tend to be positively charged. So they're going to stick to the surface of bacteria. So now they're going to stick to the surface of bacteria, release positively charged silver ions, which penetrate the bacteria, microbes, and then they're killing them. And then what happens is the silver nanoparticle gets released. It would eventually dissolve, but it's going to take a long time, and it will be killing microbes all around in the environment, which was, of course, the issue that had to be prevented. So what we did at that time was say, okay, so let's take the practically dirt particle, make the particle of desired size, functionalize it in a way so it is also going to have the positive charge that is going to make it stick to microbes and bacteria. Infuse it by physically bound adsorbed silver ions in the amount that is necessary to do the job in antimicrobial action. And then what is going to be released after the work is done is going to be just a lignin nanoparticle or like practically particle that is going to be benign for the environment. The issue of being benign for the environment for lignin is kind of understood in the sense that I mentioned that everything around, including the soil, is brown. So you don't need to kind of really look much for lignin particles and to find them in the environment. But of course, we have done tests with the EPA, which I'm going to show a bit later. So this is how the whole thing works. You have a lignin particle functionalized with a little bit of polyelectrolyte, cationic polyelectrolyte, just to change the surface charge. You have the lignin absorbing silver ions, which are then released and penetrate the bacteria. And then what happens at the end is basically just the lignin particles being released. So here is how the whole thing works and what the data and kind of the process looks like. First of all, in purely physical-chemical perspective, we need to understand how the parameters of the process affect the properties of the particle. The first thing to, of course, to control is the size. And in this case, the degree of supersaturation can be controlled by the acidity of the solution where the lignin particles are formed. So if you have more acid solution, you're going to have higher degree of supersaturation. Consecutively, you're going to have larger size particles being formed. Now, those particles are somewhere, let's say, from less than 50 nanometers to more than 150 nanometers. We call them nanoparticles for reasons that are kind of like pretty obvious for anybody who is in academics. That is not a popular terminology in industry at all these days. I mean, like it's much safer to call those colloidal particles because following that kind of like, I mean, the anxiety about potential environmental impact of nanomaterials, there has been quite a bit of pushback on anything containing nanoparticles. Even though they may be benign and so on, there is societal and industrial kind of aversion to the word of nanoparticles in products. But these are really not nanoparticles from the perspective of strict definition of nanoparticles. And then there is a very strong pH dependence, as I mentioned, depending on the type of lignin. and whether we have added the cationic polyelectrolyte or not. The cationic polyelectrolyte is obviously going to stabilize the particles in terms of the solution, but it is going to destabilize them in terms of colloidal stability. You have a certain range where the lignin particles are going to be stable and stable both in size and in coagulation perspective, right? So you have a stable suspension. It is going to be like, I mean, somewhere within the kind of like, I would say, the neutral, the mid-range of pHs. If you go to too low pH, what you see here is the particle size decreases by light scattering. But this is not that the particles become larger, they begin aggregating. If you have polyelectrolyte and to stabilize them electrostatically, you are going to destabilize them if you increase the pH, so they aggregate at high pH also. On the other hand, if you do not stabilize them with polyelectrolyte, they're going to dissolve at high pH because they are negatively charged polyelectrolyte themselves. So this is how it works. Obviously, this is not much of a problem because we can work within the right pH value. And then what happens when we have silver ions? So this is what happens. First of all, when we add silver ions to the system, lignin is known kind of as the universal sponge for anything because it will absorb any inorganic or organic material. It really vitamin good in absorption. So when we change the silver concentration into the system, more and more silver is going to be absorbed on top of the particles, or maybe they're kind of like bulk. The point is that this is a physical absorption, and as chemical engineers, we can go back to some very basic names such as Langmuir, who has been the first to study similar type of phenomena. If we can use the Langmuir absorption isotherm, it's great. So, I mean, there is no need to kind of like have anything mysterious about silver absorption of lignin. And then, when we bring those particles from the case, from the system where there is silver ions around to pure water, what is going to happen is they're going to be releasing silver. Again, they're a good chemical engineering formula which describe, and some of them were mentioned here, by the way, which describe the mass transport from a particle in the surrounding medium. So we can apply those formulas. In this case, it's a function of time, and we can see how the silver gets loaded and unloaded from those particles. So now that we know how they operate, we can also check out their colloidal stability. Again, they're a good all-time fundamental theories that describe colloidal stability, so-called D'Olbio theory. We can say, okay, so we're going to calculate the interactions based on electrostatics, repulsive, and van der Waals attractive. We can use the formulas that have been described for many, many years already. At the pH, which changes the charge, includes the role of the electrolyte, which changes the counter-ionic layer, and then calculate something like the typical D'Olbio curve shown here. Basically, this is a curve of the energy of interaction between particles as a function of the distance between the particles. This is the repulsive barrier, electrostatic, that keeps the particle stabilized if we go strictly by the D'Olbio theory. The moment this barrier goes below zero, that is, there is no repulsive interaction, the particles are going to stick together, and all the theory and the experiment kind of show that about 250 micromolar of salt, those particles will tend to agglutinate, that is, will tend to stick together, but that obviously does not need to be the case for a product which does not have that much salt. So we have a relatively stable solution, suspension of particles. We can change the charge of those particles, as I mentioned, by adding polyelectrolyte. So this is what happens in this case. You start adding polyelectrolyte and you measure the particle size just as a feedback, and you measure the charge. So the charge is going to eventually go through the so-called charge inversion, where you go from positive to negative. The size here shows kind of like unstable jump region near the zero surface charge area. So if you're making such particles, that will be for kind of like maybe our younger investigators here. If you're making such particles, make sure that you add your polyelectrolyte rapidly. So the kind of the instinct of a PhD student will be to do things, well, add the polyelectrolyte, wait a little bit, see what happens, right? And then if the things do not go well, then start adding the polyelectrolyte even more carefully in order to, well, hopefully this time do not aggregate. But really the trick here to making the system stable is to add it as fast as possible. because you have to go to the zero charge before the particles can aggregate. So now we have the particles. In terms of thinking about what will be the properties of those particles, what will be the surface charge and surface properties, the model that I mentioned was obviously insufficient to describe all the complexity of the system. So in a particle like that, you're going to have hydrophobic patches, hydrophilic patches. This is lignin, it gets bold. It is going to have ionizable charge groups, and it's going to have hydroxyl groups, which are typically the one that makes the hydrophilic patches hydrophilic. The combination of similar features is something that you can find in another type of system, which is natural systems with live cells, except that they are typically negatively charged. So now we have positive and negatively charged patches, and we are going to have the lignin particles, obviously, having affinity to cells. This is illustrated in this confocal microscopy image. What you're seeing is the confocal image of the cells, and the particles here are with another fluorescent color, which is light or white. So this is what happens. You can see that kind of obviously the particles have accumulated on top of the cells, and after they accumulate there, they're going to eject silver ions and kill them. How do they kill them? Well, we can characterize that by using essentially suspensions, cultures of E. coli and or other bacteria. We can add the lignin particles, and then we can count the number of bacteria that remain or microbes that remain after the treatment. We have done this research initially with major types of microbial cultures. E. coli, which is kind of the standard in this case, bacteria which are resistant to quaternary amine. Know that we have quaternary amine also in the system, and bacteria that are resistant to also quaternary amine, but sensitive to silver. So this is what happens. The typical characterization of such system would involve a number of controls, right? Well, it kills the bacteria, but maybe particles on their own are going to kill the bacteria as well. So what we have shown here is we have the silver equivalent of the system. We have a number of systems. The black one on the back is the one that contains silver nanoparticles, as they are highly effective antimicrobial silver nanoparticles. And then here we have different components of the system. You can have only the silver ions, only the cationic, only the lignin particles with silver but without cationic agent, and the supernatant from the particles, and then compare the Achillic efficiency, which in this case is the height of the bar here. So you can see now, kind of like, just to go to the back to the basic conclusion here, the original silver nanoparticles would kill only 5% of bacteria. The environmentally benign lignin particles are going to kill 95% of E. coli. So obviously works pretty well for this bacteria. Then we go to the other type of bacteria, which would not be killed by silver, but still doesn't like positively charged particles. So in this case, this bacteria is going to be killed by the system which contains the quaternary amine here. But the silver nanoparticles are again, well, they're maybe a little bit less efficient than the quaternary amine in solution, but still are doing the job quite well here. And finally, the third system of the bacteria which are highly sensitive to quaternary amine. So we're going to get essentially they are going to be killed by that is sensitive silver. So they're going to be killed by any form of silver, and they're going to be killed by our particles. So bottom line is, well, those particles are highly efficient for all of those microbial cultures. So now that we have a system like that, of course, you would say, okay, we want to commercialize something like this. And then, of course, you get kind of like to know that this is not so fast, right? The reason this is not so fast is because they have to be checked by agencies such as the Environmental Protection Agency. First of all, they would like to see the kind of like very qualitative level that the particles, once depleted, are not going to be killing microbes in their body. This is very easy to check. It's shown here. Basically, once we leave the particles in water for a long time, they are going to become not only not killing the bacteria, but actually help microbial outgrowth, because now the microbes like to kind of munch on the lignin particles that are present there, almost like a food. So they will kill microbes when they have silver, and if the silver leaves them, which is very easy, then they're going to become, like, obviously, naturally friendly. EPA has a much more kind of complex way of investigating those things. Kind of, they probably spend 10 times more funds on-site investigation of the particles than they gave us to make them, but that's how all research goes. So they have a very broad range of arrays, which essentially uses a number of systems and pathways, that's metabolic pathways, and finds out how the pathways in different species are going to be affected by the presence of the particles. So you can have an upregulation or downregulation of a pathway. Both of those are not good, right? I mean, you want nature to operate as it is supposed to do. So here we have the upregulation is in red, downregulation is in blue. Basically, you have all those kind of like metabolic pathways here. There's some metabolic pathways, and you can see how which ones are affected, right? Yellow color is good, blue, and the deeper the blue and the deeper the red, the worse in terms of potential ecological impact. So you can see here, if we compare the EBMPs with silver, they are like here, just the lignin particles are here, and these are the silver ions and silver nanoparticles. Obviously, the natural impact of the sustainable nanoparticles is much less. Since then, we have also worked with colleagues at Oregon State University, who have also shown similar results. Just kind of like in terms of a little bit of on the educational side, another example of how EPA evaluates similar materials. So basically, we have the so-called Cox donut type of plot. You have various concentration of the material. Again, you have the equivalent of upregulation and downregulation of different types of pathways or different cell expression processes. So basically, either of those is not good, right? It can be dark blue or dark green or light green, depending on the effect, but neither of those is good. So the less color we have on a donut like that, the better. So here is a let's say a visual comparison of the Cox donuts for the EBMPs and for silver, in this case, silver ions. So we can see that again, the EBMPs are relatively benign and then potentially much more friendly for the environment. So you have those particles, and then you want to make something out of them. You want to say, okay, so I would like to commercialize that system. Now, the problem with commercializing different types of systems containing big biomedical systems is that you're really doing biomedical research is very costly, and it's a very fuzzy area. There are many systems that can work in biomedical research. So in order to commercialize those, you need to find the right target. And we have identified the right target, in this case being agriculture, as having much easier, let's say, much lower threshold for making the system work and much broader application. Now, if you want to make something like an agricultural application, so biomedical applications are good because you only have to make a small volume of highly valuable material. But if you want to use an agricultural application, now we have to make a large volume of, well, relatively inexpensive material. So how do you make such large volumes of particles in a way that makes financial sense, if you will? I mentioned what people think about lignin. Now, of course, there are those methods which are very well known in industry, so-called impinging jets, where you mix two streams at very high velocity. You have very uniform particles. This is not the problem. And mixing streams at high volume fraction, at high volumes is not really what you want to do in an even small company environment, because you don't have that much liquid and space to operate large throughput systems. You want to make highly concentrated system of particles. So you make something like the thing shown here. You are going to inject lignin solution into a flowing liquid, and you're going to get particles. But you're not going to get high concentration of particles. It's a relatively low concentration. Now, the instinct in this case is, well, let me increase the concentration of lignin. But when you increase the concentration of lignin, what happens? For those of you who are taking this educational opportunity, you should remember, what happens if you increase the lignin concentration? Yeah, so this is not what you want, right? I mean, you don't want your particles to become too large. So what do you do? Well, I mean, what we have found out, and kind of this has been described before, but in this case it worked beautifully for our system, is that you can recirculate the medium. So basically you keep injecting lignin and then you add lignin into the system where you already have lignin nanoparticles, but you keep precipitating them. And then you find out what happens. And what happens, like, well, first of all, you find out what happens if you try to increase the concentration of lignin. Particle size increases, right? Not what you want. I mean, you may want to stay within a certain size, so the particle size would increase in concentration. You are going to destabilize the system eventually, so that's not the useful type of process that you want. But then, if you do a kind of characterization of the size of the particles in a system with recirculation, and in this case, we have this kind of these days we are doing this for a variety of colloidal stability system. So we take the light scattering plot, and then we plot it over time in order to get this three-dimensional time-dependent plot. So you're immediately seeing here the size distribution, and some of the similar type of plots were shown here for aerosols, I think. So you're following the size distribution over time, and you are checking out how it changes. And what you find out here is actually the size distribution does not change over time in the system of recirculation. So that's kind of surprising because originally the idea was, well, you know, if you add more lignin, you get larger particles. But in this case, you keep adding lignin to the system that has already lignin particles, but the size distribution does not change, even though the concentration of particle changes. So this is very useful because you keep adding particles of the same size under the same conditions. The reason for this type of behavior is so-called non-Lamerian nucleation. So the typical way of making particles, which has been described again from the time of Victor Lamer, is basically if you have nuclei, those nuclei are going to keep growing. But in biopolymer systems, you can have the case where every new material that you add to the system is going to lead to new nuclei. They're going to grow to a certain level, and then because of the recirculation, you take them away and you add more solution, and you keep adding lignin, and eventually you get up to the side to the case where you have this kind of uniform distribution over time, which is very good because now we can intensify the process without scaling up the system. We scale up the number of recirculations that the particles have to go. So we have this process now. If you want to prove something like that, I mean, you can, of course, play up with the system parameters. You can kind of do, in this case, a hypothesis, which is the following. If this is exactly the case, you can hypothesize that if you add lignin consecutively in the system, you can do this in one flow even. So first you have, let's say, lignin of larger concentration, and then lignin of smaller concentration. They should produce lignin particles of different sizes. So if you do that, you should have at the end two discrete different distribution of the particles that go into one of the same stream coming out. And in this, when you do the checking of the system, you really are going to have one particle. If you go and check out what happens after the first injection site, you have one size of particles. If you check out what happens after the second injection, you have two concrete particle distribution, very nice, very narrow. I'm not sure what exactly would be the use of two size distributions at the same time, but if you want to do it, at least it proves our hypothesis. And I mentioned that we can now use similar particles and scale up the process, and you can use it for different types of commercial applications in agriculture. How many minutes do I have? It's kind of simple. My time is over.
Yeah, we started 5 minutes late, so 5 minutes left.
Maybe I can give you a little bit of an idea of now. So now you want to commercialize something like that commercially. Biomedical is expensive, long, and tricky. And agricultural is something that people would like to kind of like really find replacement. So the big deal about agriculture is that you're spending lots of synthetic chemicals. And those synthetic chemicals, typically you put them in some surfactant solution and you spray them on the leaves, but some of it will drop off the leaves into the ground. And then next time you have rain, you have surfactant and it's going to self-wash itself and still going to the ground. So it is not going to be very efficient. So one of the concepts that has been emerging, and luckily we have been ahead of the wave in this case, is if you take lignin nanoparticles, you really have the means of delivering something onto the leaf that is going to stick to the leaf, and that can keep the active ingredient there. So we started kind of like, basically this commercialization process with the startup company, small startup company, where we replicated this process of continuous circulation in kind of like really very small space. I mean, you can generate lots of lignin nanoparticle suspensions. We can functionalize them with chitosan in this case, not with polyelectrolyte, because polyelectrolyte is synthetic chemical. So it replaces the synthetic chemical with kind of natural polyelectrolyte, positive polyelectrolyte, which is chitosan. We do not need silver for similar applications. We can kind of like use the particles as they are because they already lignin and chitosan have antimicrobial and antifungal properties. And then what do you do if you have a system like that? Where do you go? Well, you go to the state of North Carolina, but you also go to the state of Florida. And here in Florida, it turns out that there is a so-called tomato growing association, kind of like very, really large and kind of kind of like a connected group, which would like to see similar type of developments. So my colleagues at the company have pretty much made samples and investigated them in Florida, in North Carolina. And they have found that those formulations are nice and efficient as replacement of synthetic products that are otherwise used in agriculture. So here is an example. What we have here is, this is the so-called area under the disease pressure curve. Not my area, not my area of research. But I mean, like you can imagine what it means, and basically a lower value means better result, right? So I mean, this means that less disease in this case in terms of tomato, bacterial spot of tomato. So if you put water, I mean, you don't get much. Some chemicals actually are not effective at all, just because the fungi have developed and bacteria have developed resistance to them. There are some chemicals which are kind of like the so-called chemical control these days that are efficient, but are synthetic chemicals. And the nanoparticle material is about as efficient as the best synthetic chemical that's chemical control here for our standard. And then there are other formulations which are more or less efficient, but our formulation is about as efficient as the best chemical that you can get from companies such as DuPont a few years ago. You can mix those systems and you're going to find out that they are synergistically. So basically the nanoparticles are not an alternative to chemical treatments. Actually, if you mix them, you're kind of like having a system which is more efficient than the particles and the chemical themselves, just because you're really attacking the disease by two very different mechanisms and means. So potentially that is the way to go. I'm not going to read all of this. I mean, you can probably imagine this is kind of like the company description of what the technology looks like. So obviously there is lots of potential here. These are only first stages. There is going to be quite a bit of further development. I have a few slides which relate to making particles of special sizes, but I'm going to just skip through those, kind of like just mention very briefly what we do. We can make particles of different sizes. We can make tubular particles in liquid medium and then dry them out. We can make them from biopolymers such as chitosan and lignin. Lignin rods are not kind of like specifically right now something that we have an application, but chitosan nanofibrils are very good in one of the potential applications, by the way, is capturing aerosol. So if you take a textile filter and if you coat it with similar type of particles, you're going to have, this is the figure of particles that we make, you're going to have a very efficient, you increase the efficiency of the filtration for aerosols. You can have different types of super hydrophobic coatings. Going back to sustainability, you want to use similar biopolymer to capture something that we don't want synthetic, which is microplastics. And those particles make great ways of capturing microplastics. So basically, kind of sustainable material are not only a replacement, but in many cases, solution to different problems that are facing society because of other types of technologies. So hopefully there's going to be lots of interesting developments in this area. Of course, we are kind of like just one of the many players, and you're going to hear lots of interesting developments from other groups, I'm sure. I kind of like, I mean, this is a summary of what I have been mentioning. I'm not going to read through all of that. I would like to acknowledge again the people who have contributed to this work and also extend a special invitation. I have been very busy, as I mentioned, with some of you organizing the ACS Kuwait Symposium this year, which is going to be in June. North Carolina State University, one single direct flight. So please join us for this symposium. And again, thank you very much for this opportunity to tell about this type of materials.
Thanks so much, Alan, for the excellent. Deep, can you try to? Yeah, hi, Deep, can you try to? Is he going to project the slides? Yes. Yes. Okay. So Deep, if you can share your slides. Deep, can you?
Yep, I sent my slides this morning. I don't know whether Professor Yang had received them.
Oh, now we can hear you very nice. Can you project it?
Yeah, but I think maybe in terms of controlling the slides, it's better that you share those things, when you project. When you see.
So Deep, are you sharing your slides?
No, I think I emailed them to Yang. So should I share it from my?
Yeah, I think it's better to share it so that you can actually go through the slides as they go through.
Okay, no problems.
And then can we keep up on the screen as well, or that one is still showing the old one?
One minute.
We're just waiting for a few minutes just to kind of get this organized.
Can you let me know if you can see my desktop?
You should have shared that. Not yet.
Okay.
You maximize that. Yeah.
How about now?
No, not yet.
Oh. Because I'm sharing my screen.
Where is his slides? The one he sent us.
Okay, can you see it now?
Yeah, almost.
Okay, yeah, now we can see it.
Let me put it on there, right? Good.
Yeah, that's great. Okay, so you can hear us, Deep, right?
Yep.
Oh, yeah. That's great, everyone. So it's a pleasure to introduce Deep Bhattacharya today. Deep is currently the Vice President of Care Solutions Technology and Technology Design at Eastman Chemicals. He has been with Eastman for the last 20 years. And he has had several positions in the technology and marketing organizations. Prior to his current role, he was Director of Global Coatings Technology. Deep has a Bachelor of Science degrees in Chemistry and Chemical Engineering from India and a Doctorate of Philosophy in Polymer Science from the State University of New York. He has several patents which he has filed throughout his career and serves on various committees and editorial boards of various journals in the coatings and other broader areas as well. So he's going to talk to us about sustainability and circularity-driven innovation at Eastman. Again, Deep, thank you so much for today. And I'm going to hand it over to you for the talk.
Thanks, Amil. So just in terms of housekeeping, how much time would I have for this? Just want to be mindful of 15 minutes.
Yeah.
Okay. And appreciate the invite. I was hoping to be there in person, but some work-related challenges had me, stay put in Kingsport. That's where we are located. But what I'll do in the next 15 minutes is talk you through two of the themes that are really important for us at Eastman in terms of how we look at innovation. And those are based on sustainability and circularity. And what they exactly refer to as, we look at it from the Eastman lens, I'll touch upon that, those two topics pretty quickly. Can someone help me? Okay, I think I've moved it. Yes. Can you see the next slide, just for folks in the room?
Yeah, you can see.
Okay. So just for some context, Eastman is a 100-year young company. We had our centennial 2 years ago. So it was founded in 1920 by George Eastman. We continue to look at new ways of innovation, but what has been more important in the last decade or so is a much more stronger focus on sustainability. And we look at it through 3 different vectors. The first one is linked to mitigating climate change. The second one is, how do you bring sustainability in terms of an overlap with mainstream circularity? And I'll talk to you about what that refers to. And the last piece is a very strong commitment to caring for people and the society. So there are 2 big programs within Eastman right now that I want to sort of share with you, that looks at sustainability and circularity and the overall impact on the material space within our society. So if I go to the next slide, let me see if you can. Oh, I don't see that advance. Samir, can you help me move to the next slide, or I know how I?
We're kind of seeing again. I don't know.
Let me.
So if I think you are sharing your slides right now, so we can take it on your slide, as that proceed.
Sharing, and I'm advancing it on my laptop, but I somehow don't see this.
I mean, the other way is just project his slide. I'll change the slide.
Sure. But then we should download this.
I apologize, Samil, for the inconvenience. Yeah, it's a one after this.
Yes.
Have you moved the slide? Because I still see slide number 2 on the screen.
So we're trying to kind of get the slides you sent, and I'll try to change it manually here.
Okay. I don't know, why is there a?
It's almost download.
Excellent.
So this is our slide.
So I can manually, if you tell me to change the slide, I'll change it.
Thank you, Samuel. Yeah, so let's just move to the next slide.
Okay, we're on the third slide.
Yes, thank you. So this is one of the biggest efforts within Eastman to tackle a significant challenge for both the developed as well as the developing regions, and that's linked to plastic waste. And there is a lot of bad rep that the plastic industry gets from environmentalists about, the fact that it's not linked with sustainability goals, but the story is not entirely explained clearly to the consumers. Plastics are an essential part of our lives. You can like it or you can dislike it, but fact remains that is the most efficient way to transport things like potable water, packaged foods. It's even at the leading edge of helping us advance some of the medical technologies. So, fact that plastics play such a pivotal role in our lives is something that we need to, embrace and accept, because otherwise it would lead to significant material challenges. People talk of glass being an alternative, but it has a 4 times the same carbon footprint as plastic. And imagining, transporting water to the hinterlands, to the difficult parts of the world in glass containers compared to plastic creates a really challenge on the overall CO2 footprint when it comes to the transport costs and the transport efforts to ship products like those. Therefore, the question is not how we look at plastics in terms of the life cycle, but are there ways to reduce, reuse, and recycle plastic? And how can we make a technology that's more circular, where you're taking something that's from an oil-based source, but creating an infinite loop? And that's something I'll touch upon really quickly, because it's one of the big moonshots that we have at Eastman right now, is to create that circular technologies that will help the polyester segment per se. So if you move to the next slide, Samuel. Yeah. And this is just in terms of context, if there's something you can take home today from our discussion, is there is a significant opportunity that presents us, people, engineers and chemists and scientists working in the material world. There's about 300 million tons of plastic that's produced annually. And this is how it splits down. Out of the 300 million tons, you have about 40%, a little bit more, that goes to the landfills, both in the US, but more recently in the emerging markets. Initially, it was the Southeast Asian region. Now it's more in Africa. So about half of that goes into landfills. And you've got another 25% that's incinerated. So those of us in the developed regions, we hear a lot about recycled plastics. But when you take a closer look at the numbers, about 16% of these plastics that are produced annually is actually meant for recycle, or there are provisions in place that allow you to have an ecosystem based on mechanical recycling. But out of the 16%, only 12% actually goes through a recycling process. So when you do the math, you've got 300 million tons of plastic that are produced globally. And in reality, only 3 million tons of that plastic is actually being recycled through the mechanical recycling process. That's 1% of the annual plastic production. So this creates a significant challenge, as well as a tremendous opportunity to find ways how you can supplement and complement the mechanical recycling that exists today, and create more technologies that will allow you to get around having these landfills get more and more accumulated with plastics that serve as, for example, for single source applications. Right. So at Eastman, we've spent the last few years to look at different ways of, is there an approach you can basically unzip a polymer into its constituents and make those building blocks accessible to creating new materials in the design space. So Samuel, if you go to the next slide. Yeah, and this the problem that we are trying to tackle is, when you look at the PET industry, when you make virgin PET, for example, when you buy a Coke bottle from a dispenser, or you're looking at a water bottle at a gas station, that's the clear PET. Now you hear that all of that gets recycled, but there are limitations to not just the overall ecosystem, but even to the overall material properties. So as you go through each round of iteration, as you recycle clear PET, there is an eventual breakdown in properties. And what you see through this funnel is the clear PET over time moves into a downcycling of applications. It goes from clear PET to things that are more opaque, eventually gets to colored PET applications, and finally it becomes used in areas like carpets, as well as in the design of fabrics. So over time, it's not actually an infinite loop. If you look at the actual iteration process, you can get probably 3 to 4 rounds of recycling through a clear PET polymer that's produced in the industry. And as I said, only 1% of that is actually getting recycled today. So the way you can tackle this challenge is what we refer to as chemical recycling. And by that, I mean you can take PET, and once you have access to the PET that gets discarded, you can actually break it down into its constituent monomers. So Samuel, if you go to the next slide, That's the chemistry I'm talking about. So if you take PET in the presence of methanol, think of a Lego block. You can actually break it open into the individual building blocks. The two building blocks are DMT and EG, and that allows you to start off with the monomers that was initially used to make the virgin PET and thereby create an infinite loop. And the good news over here is once you've got this ecosystem established, there's no really trade-off or downcycling in properties as you go through this chemical recycling process. So that creates tremendous opportunities for us to look at current areas where there is a high dominance of PET being used, and can we create a system where you have this infinite loop? We've signed MOUs with Pepsi, with Procter and Gamble, with the French government, where we are actually building the world's largest chemical recycling plant. And the goal over here is to actually recycle 250 million pounds of plastic on an annual basis. So if you go to the next slide, this is a very exciting year for us because we are in the process of actually scaling up and commercializing a full-scale plant at our headquarters in Kingsport, where the goal is to actually move towards processing this level of feedstock by 2025. Now, the question is, you may ask, is what does 250 million pounds of plastic look like? I haven't been to the Gator Stadium, but if you look at another example for, from a context standpoint, if you go to the next slide, Samuel, in Tennessee, the Vols are a favorite team, and the Neyland Stadium in full capacity holds about 120,000 people. So when you think of 250 million pounds of waste plastic, that is enough to fill the stadium 2 times over. So that's the volume of material we're looking at. And the goal is to start processing that feedstock starting this summer. So clearly a very exciting time for us in terms of where we are from a sustainability goal in looking at, taking plastic that's available in the market today, breaking it down into the individual components, and then scaling them back to other value-added applications, depending on what the market demands are. So that's one example. I'll very quickly touch upon the other one, Samuel, and that's linked to microplastics. Olive, he referred to that situation in his earlier presentation, where that is a growing concern. If you go to the next slide, Samuel, this is a significant issue that has come into being over the last 20 years. And when you think of the microplastic challenge, there's about 42,000 tons of microplastics. So about 100 million pounds of that being consumed just for the European segment today in the personal care space. Now, when I look at the global usage, that'll be in excess of 300 million pounds. And unfortunately, a lot of these applications where microplastics are used, they are in the B2C segment where consumers are heavily exposed to microplastics on a daily basis in a lot of the personal care products. So those find home in a lot of products we use on a daily basis, be it a skin foundation, be it a lip gloss, be it an exfoliant, because they do serve some purpose both from visual, aesthetic, as well as functional areas. But the challenge is once you wash them, they go through the water system within our the sewage and end up in the oceans and in the rivers where they don't biodegrade, and that has become a significant issue. So the challenge right now is, can you replace these microbeads that are less than about 10 microns in size, which are traditionally obtained from nylon 12 and PMMA? That's become the sort of the high runners in the personal care space. Is can you find a way to replace these microbeads from oil-based sources into more bio-based options that eventually biodegrade? And the way we are looking at it, if you go to the next slide, Samuel, is there is a societal pressure where people really want to be more conscious in terms of how they look at products they buy on a day-to-day basis, especially in the personal care space. There are regulations coming into effect. So the ECHA has actually banned the use of microplastics in some applications. Plus, we feel that a bio-based source is just the right thing to do, because then you're managing the entire ecosystem in terms of how you're sourcing the product and how it ends up being in a situation where it eventually biodegrades. So as you can imagine, the two big vectors that you look at for being successful in this space is you want something that's bio-based. The previous talk focused on chitosan as well as on lignin. Cellulose is the most important biopolymer or the largest available biopolymer, and that's what we use as our feedstock. And then you basically perform some chemistry on it and eventually functionalize it. But the goal over here is to make sure that once it delivers the functionality in the formulated product, it'll eventually biodegrade once you seize that application. So that's where we are today. There's a lot of work going on. I've been fortunate to collaborate with Samuel and his team when he was at his previous assignment in New York. But I was looking forward to sort of visiting your center this time over. But I'll be there hopefully in Q1 to understand how we can collaborate on this topic. But we have made microbeads that are less than 10 microns in size. There are three really important aspects to actually launching them in the personal care space. They need to biodegrade. They need to have the performance that you see today in nylon 12 and PMMA. Some of them being the visual and the aesthetic, as well as in terms of oil uptake being another requirement. So that's a work in progress right now. We're moving full steam ahead. And the goal is to actually get the leading formulators like a Chanel, like a L'Oreal, like an Estee Lauder, to start replacing what they have today in terms of their current usage and moving towards and transitioning towards more bio-based products. So that in a nutshell is what I wanted to cover today. A little bit over time, Samuel, but just wanted to give the audience a flavor of where we are in terms of sustainability and looking at circularity at Eastman.
All right, thank you so much again. We really appreciate it. You can just stay around for the final discussion.
Sure, I'll be happy to.
So, while Yang is setting up, I'll introduce our last speaker for today. So it's Dr. Divyandu Mukherjee. He's a research assistant professor in the Department of Chemical and Biomolecular Engineering at the University of Tennessee, Knoxville, where he directs the Nanobiomaterials Lab for the Energy, Energetics, and Environment area. His research focuses on the synthesis, assembly, and characterization of functional nanobiomaterials with transformative performances for energy, energetic, and environmental applications. So Dr. Mukherjee received his BE and MS degrees from mechanical engineering from University of Baroda, India, and the University of Buffalo, SUNY. He earned his PhD in mechanical engineering from the University of Minnesota. Prior to his current position, he was a postdoc at the Oak Ridge National Labs and a Gibson Research Fellow at UT. So with that introduction, again, thank you so much for your time today to kind of give this talk. And Dr. Mukherjee will be talking to us about reactive laser ablation synthesis and solution, a new paradigm for synthesizing functional composite nanomaterials. Thank you so much.
Introduction and thank you for the organizers for the invite for this wonderful workshop. Okay. And it's been a pleasure meeting you all, and that's the last session. So thank you for sticking around, and it's shows your commitment to the CAST. And I look forward to it being the last session. I look forward to many more such workshops with CAST in future and collaborations. So as the introduction went, I did not have to repeat the first slide. It's the reactive laser ablation synthesis and solution is what I'm talking about, and for the synthesis of composite nanomaterials. So in terms of reactive laser ablation synthesis, to understand it, it's laser ablation is essentially using high energy laser plasmas to synthesize nanomaterials. That's been known in the plasma community. Now you're bringing that and interfacing it with the reactive part is where you interface it with solvent chemistry or solution to bring the two worlds together. So one, you have the laser ablation, the top-down approach, and then you get the solvent chemistry or wet chemistry that has been well established again. So the goal was to get greedy, and can we bring both of them together to do the top-down and bottom-up approach and interface them simultaneously?
Make sure that you're sharing. Share this time.
Okay, so yeah, so on that note, now this is being an aerosol, my original background. Okay, so my original background is in aerosols. My PhD at the University of Minnesota with Professor Mike Zacher. I worked in aerosols, specifically with aluminum nanoparticles for energetic nanomaterials. So in terms of the aerosols, I'll start off with okay, here you go. The gaseous synthesis of nanomaterials in the aerosol route. Something that I was doing back in my PhD is I'll start off with that, and I don't need much description for some of these processes, which Professor Biswas gave a very good tutorial yesterday. So the nucleation from monomers of gas or vapor and metal vapors, forming the first nanoparticles, the critical clusters, as they call it, the critical particles, and then they grow to surface growth, and then they can undergo two different pathways in terms of the aerosol route. One is in which you have collisions which are faster than coalescence, in which case you form these fractal-like structures or aggregates, agglomerates, with little nicking. But then these have, due to the large heat loss, they have surface oxidation. So I was looking at all of them: the coagulation, coalescence, surface growth, through evaporation, condensation, and then surface oxidation. Whereas other route is where you have enhanced oxidation, where the coalescence is faster than the collision, in which case you have large sintering and you have these neck formations. And the neck formations can then lead to like fully sintered particles, where you lose the surface area, the fractal-like structures, and the heat release from the surface energy compensation as a surface area reduction. You can imagine the coalescence, a surface energy reduction will have to be compensated by bulk energy increase to conserve the energy, in which case there will be a large temperature. It will be a non-isothermal sintering process. And that will then promote further oxidation of the surfaces. So you have enhanced oxidation. And somewhere in between, you have controlled oxidation of like neck formation of these agglomerates or aggregates, sorry, or you have these thick shell oxide shells on the particles. Now, these are, of course, these become the two pathways lead to two different formations, where you have one that can form these fractal-like structures, high surface area, more reactive, where the core metal is preserved. We're looking at aluminum for energetic activities. And then you have this, on the other hand, you have this fully oxidized or largely oxidized surfaces, where you have spherical structures with low surface area, and then those are less reactive. So back in the days, I was doing kinetic Monte Carlo simulations. So actually, and then I revisited it recently in my own academic career, my own faculty career, where we looked at the Gibbs free-energy-driven kinetic Monte Carlo simulations to look at nucleation without capillarity approximation, which means going beyond the classical nucleation theory, where the surface tension is size dependent. And then for the comprehensive kinetic Monte Carlo model, we also looked at the coagulation-driven kinetic Monte Carlo, where we're looking at coagulation, coalescence, sintering, and surface oxidations. And that again, it was these were my PhD work, and later on I extended that code to include the surface oxidation part to look at how that non-isothermal coalescence actually affects the oxidation of these metal nanoparticles during the gas phase synthesis. What we observed was, and this was the first time study to account for that non-isothermal effect, that there is heat release when the particles coalesce, that there will be a large heat release. In fact, the temperature rise was significant due to that. And if you look at the entire code, the results, the final results, there are these two peaks. The first is where you get these nanoparticles which are formed. You start in 1 nanometer particles. They form this oxide shell. During the oxidation, this is the first large heat release, the first peak. Then there's a trough. The trough is because of the diffusion limited oxidation. And I'll come back to that later on when in my recent work, I tried to overcome this diffusion limited oxidation for energetic nanomaterials. That diffusion limited oxidation, the oxygen has to go through this shell, which slows down the oxidation, so you have lesser heat release. But then the particles being still very small, less than 10 nanometers, they undergo coalescence. And coalescence releases heat to temperature increase of the particle. Then the particle temperature again increases till it becomes large enough that it starts losing heat, and then it starts the aggregation. This pathway starts taking over, where there's large number of collisions, and they start growing into fractal-like structures, and they cool down because of large surface area. But the whole point is that there are two things which is critical. One is that this peak wouldn't have been there if there was no non-isothermal coalescence effect. And the other is that in the energetic community, I don't want this trough. What I want is the entire energy being boom released in one shot. So this is basically costing when it comes to making solid propellants in rocket propulsion. So the aerosol manufacturing process, I took this from a review from Proximus' group. The aerosol manufacturing processes, if you think about it, it consists of the reactor, where you synthesize these particles, and you cool down, collection, storage into silos or hoppers, and starting with the raw materials. Raw materials could be liquid precursors, could be solid targets, and if you're doing a furnace reactor, or there's some, but it's not much done with in the case of solid gas precursor, solid gas phase precursors. And then the secondary pathways, which are these hollow arrows, are once it is formed, then you need a secondary process to coat them or functionalize them, and then you can bring them back to the collection process. So these are all like the pathways are all correlated in the way it's given. What is important is that in the aerosol pathway, you need the secondary, this coating and functionalization is typically done with the wet chemistry approach. So there's still work going on to make a single step aerosol process in which you can do the entire thing, right? And one of them would be that you have these secondary coating or functionalization precursors also in the aerosol phase itself, instead of doing it in the wet chemistry approach. And then finally, you can recycle the gas, recycling, you can do the gas treatment to take the byproducts, which can be solid or the gaseous byproducts from it, and then recycle it to the raw materials, to the original precursors with some treatments, and put it back in the reactor. The reactors, which are known, a few of them that I have covered here, spray or combustion synthesis, and then the furnace aerosol reactors, where the flame spray, you have the precursors and an diffusion flame with which you make these synthesized nanoparticles, or in the furnace aerosol reactor, you can start with the target and then evaporate it and then condense it. This was extensively done by the Proximus and Biswas group. Then there is the non-thermal plasma synthesis route, which is done by Coates-Huggins group, where they use RF plasmas. These are low temperature plasmas with which they can make nanoparticles. And in the plasma sector, there is another one, which is the high energy or high temperature plasmas, where you have arc discharge synthesis or laser ablation synthesis, both of which was done with Mike Zacharya's group during my PhD. I had worked on them, especially the laser ablation, but this was gas phase laser ablation that I was doing. But this was the original seed of idea with which I started. So always you get some idea during the end of your PhD. And the idea was that, okay, if I can do it in gas phase, can I bring the best of the two worlds and do something as a reactive laser ablation where I can do it inside a liquid, make the plasma inside a liquid, so I can get both the advantages of the high energy plasma synthesis as well as the wet chemistry. So this brought me to one of the reactive laser ablation synthesis, which was the reactive part was galvanic replacement reaction in solution. We've patented it now, and the third patent is now coming out. And the idea was that you have a target inside a solvent which has the precursors loaded in it. You choose the precursors for the chemistry that you want. And then you have, those are the precursor molecules. You have pulse laser, forms a plasma plume. The plasma plume expands, creates an ultrasonic shock wave. The shock wave expands into the liquid, creates a cavitation bubble. Inside the cavitation bubble, the CD nanoparticles are formed from the melting of the laser ablation process. And those nanoparticles will then be thrown through the cavitation bubble into the liquid bubble interface, where these nanoparticles, seeding nanoparticles become the sites, reaction sites, or the secondary reactions, chemical reactions in the solution. And with that, we can make a large number of nanocomposites, nanoalloys, core-shell structures. And the two, as I said, there are two disruptive processes which are coming together: the laser ablation synthesis inside the plume or bubble, and the solvent chemistry, or the galvanic replacement reaction, or depending on the solvent that you choose, you can have laser-induced pyrolysis, or I'll show you some results where we have made moths. So doing the imidazole chemistry, and you can, though that's happening at the bubble solvent interface, where the nanoparticles are thrown into the liquid. The parameters that we can use, that we have multiple knobs that we can turn around, both on the solvent side and laser side, as listed out here, which includes laser energy, laser properties, and solvent types of solvent, precursor concentrations, temperature, pressure, pH, etc. What are the merits? I've already said many times: disruptive top-down lasses with bottom-up solvent chemistry. We are bringing a disruptive merger of the two. One pot, one-step synthesis. We are able to get high energy non-equilibrium process. We have been able to make metastable structures because of the high temperature and pressure under which these materials are being formed. Low aggregation from charge screening effects, structure composition controls using all these solvent and laser parameter knobs that we can turn around, and then low wall losses. So you have high collection as low density salts. Now, talking about aerosols, this belongs to the colloidal family, but more to the sol family. And that leads to scalable nano manufacturing. Demerits? Well, although the low aggregation can create a pretty small dispersion of the particle size, but there is still what we find out because of the very dirty process, the very high energy laser process, this weak control of the particle dispersion and the morphology, particle size dispersion and morphology. It's a complex process. Now, that is, I think, of course, it's demerit. Being an engineer, the best design of a process is simplicity. How simply can you make something? If you make it too complex, that's not good enough. So this is one problem, is it too complex? So it heavily depends on how creative you are in the design of experiments. Each design, you have to choose the right solvent, choose the right target, what energy do you use? So it's not like push the button and get it. Non-equilibrium process belongs to both merit and demerit because it becomes challenging to do the plasma diagnostics. Most plasma diagnostics, plasma physics, like Maxwell-Boltzmann distribution, Shah ionization, Shah-Boltzmann distribution, they're all based on local thermodynamic equilibrium. Cannot employ any of them here to analyze this plasma. So looking at reaction kinetics from the plasma, it becomes challenging. And then there are artifacts due to the laser shielding from this cavitation bubble, when this laser interacts with the cavitation bubble, which creates some productivity problem. What did we make? We made a diverse world of functional nanomaterials using our analysis techniques since I started as a faculty. Those are some of the papers, and we have used them for making fuels electrocatalysts for energy conversion, supercapacitors for energy storage, solid propellant applications, which is for energetic additives. To highlight, recently we have been able to make MOFs using this laser ablation technique, reactive laser ablation, with the solvent chemistry was the imidazole chemistry to coordinate metal ions from the plasma with imidazole to make MOFs. And then we turned these MOFs, which is a second step, not a one step, but then we parallelize them to form highly porous, ultra-porous carbon structures in which these platinum cobalt nanoalloys were being formed, which were found to perform extremely good OR electrocatalytic activities. And in recent years, as I said, in our work with defense, we have been able to make a unique metastable nanostructure. We have been able to trap it for the first time. Unfortunately, it's a limited distribution classified material, so I will not cover it. It's under distribution D, but all I can say is that it has shown some very promising performance with the Army. But today I will be talking about two specific areas, electrocatalytic application and then the energetic applications. I just showcase what have we been able to do. So I'll take two cases of materials that we have made. One is this platinum cobalt nanoalloys embedded in a porous cobalt oxide bed as nanocomposite, and the other is a graphitic shell-coated aluminum nanoparticles as energetic materials. The first one, the platinum cobalt oxide nanocomposites. This is for OR electrocatalysts in alkaline conditions. The last is GRR technique that we employed. Cobalt was the target, and then we're using laser ablation that produced the cobalt seeding nanoparticles, which then underwent redox reactions with the potassium chloroplatinate salt, which was in the solution, to undergo galvanic replacement reaction to produce platinum and cobalt ions, which then the excess platinum goes and alloys with the cobalt to form platinum cobalt alloy, and the excess cobalt ions form the cobalt oxide bed or the matrix in which these platinum cobalt nanoalloys are embedded that prevents aggregation. It's driven by the redox gradient. As you can see, the negative redox gradient of cobalt to cobalt 2 plus as compared to the highly positive redox gradient of platinum reduction of platinum salt. So hence this goes and reduces the platinum salt. What did we produce? I'll showcase one of the samples. These were the platinum cobalt nanoalloy particles, sizes of around 8 to 10 nanometers, very small, pretty good distribution. We looked into the detailed high resolution TEM. These were the platinum cobalt 111 alloys formed with the D-spacing as shown there. And then the rest were cobalt oxide as shown here. And there were two different, the cobaltosic and cobalt oxide, CO304 and COO, and that's why we call it COOX. The two different kinds of oxide that were formed. EDX showed that these were indeed, inside there was a platinum cobalt with not much of oxygen. Outside there was no platinum, but cobalt and oxygen. What did they do when we did the electrochemical characterizations? We did, as we said, in alkaline conditions, 100 pOH, rotating disc electrode measurements. This particular sample, which had 1:2 molar ratio of platinum cobalt, any higher, these had lower platinum content, didn't perform as well. as well as when we went to higher platinum content didn't perform, but the ideal composition of 1:2 molar ratio that showed an activity, the electrocatalytic activity, which was higher, which is the current per unit mass loading of platinum, higher than the standard commercial platinum, which are currently used as OR electrocatalysts. When we look at the overpotentials, which is essentially compared to the oxygen reduction to water 1.23 volts, the overpotential above it, calculated from the halfway potential of the cyclic voltammetric curve. Then I'll just not get into the details, but the combined overpotential, this is the ORR overpotentials, this is the OER overpotential. The lower the overpotential, the better the performance of an electrocatalyst. And if you see that this platinum cobalt for both combined ORR and OER had the lowest overpotential that we could achieve. Stability as compared to platinum, the stabilities of this, you can see this is cobalt oxide, pure cobalt oxide, standard platinum, commercial platinum currently used for electrocatalysts. And this is the blue one, I'm sorry. And the red one is our alloys. And the stability of these over 12,000 seconds were much better than the commercial platinum catalysts. So why did this happen? The reason is you have platinum cobalt alloy, which causes lattice shrinkage, allows the 5d orbital vacancies for the platinum oxygen bonding, which allows here that you have the oxygen absorption onto the platinum cobalt alloy, and preferably due to the lattice shrinkage, whereas the desorption of the OH, it promotes the oxygen absorption and desorption of the OH, and the OH then is taken over by the cobalt oxide matrix, which is the porous, highly porous, spongy cobalt oxide matrix, which then promotes a synergistic activity for it, thereby giving over this, what's called in this community as spillover effects, where you have oxygen preferentially absorbing to the platinum cobalt alloy and the OH minus, which are being dissolved into the cobalt oxide, which then undergoes OER. So simultaneously both OER and ORR. And furthermore, these cobalt oxide bed prevents the aggregation of these particles. In terms of the energetics, I'll just quickly go over this. The problem, as I said, was the diffusion limited. When I started working with Mike Zachariah, the diffusion limited oxidation in aluminum nanoparticles with oxide shell formation. The basic concept was, well, if that is the problem, then coat that aluminum with a graphitic shell on it. The idea is that if you have a charcoal for your barbecue, we normally put the fuel on top of it. Imagine if you had a way in which you had the charcoal and the fuel is inside. And that's what I exactly described to my grad student. And then I realized that it was dangerous. And I told them, don't try to mark your Friday barbecue. But the graphitic shell coated prevents it from oxide shell formation. It hinders the diffusion limited oxidation. Carbon, when it burns, only forms gaseous products. There is no ash formation. So there is no shell on it. And then furthermore, the graphitic shell will hinder any porcelain. This time we did aluminum target in toluene and acetone to pyrolyze the acetone and toluene and created these aluminum coated with graphitic shells. High resolution TM showing aluminum at the core, carbon graphitic shells at the edge. And under toluene, they formed aluminum carbide crystalline structures at the core. There are two different forms of energetic materials that were formed. We sent these to the Army lab. Collaborator Dr. Jennifer Gottfried did with her patented technique called the laser-induced air shock from energetic materials. They tested it. And this was basically you blast these materials and look at the shock wave propagation, and from there you get the definition velocity or shock velocity from these. The results turned out to be, here it is. This is the commercial aluminum nanoparticle that they're using currently. These are our materials that have been the graphitic shell coated that have been formed in acetone. And these are the results from the Army lab tests. And again, the aluminum, the carbon coated aluminum also showed fast kinetics, the blue line, whereas the aluminum carbide carbon coated showed slow kinetics, but large heat release over a long period of time. So we can control the fast heat release or large heat release by tuning the structure and composition of these materials. The funding support from Army, I'd like to acknowledge from Army, Air Force, NSF, and the Navy. I also recently got the 4521, the DURIP, for transforming this lasses that I have now into a machine learning driven large scale manufacturing platform so that we can do large scale manufacturing energy materials. My collaborators I'd like to acknowledge Professor Komami, Professor Dusher, Professor Zalzalski, and Dr. Jennifer Gottfried from Army Lab. That's the students' work. These are the students who make it happen. All the results are presented by Shenku, Davari, Ali Davari, Eric Ribeiro. They graduated. This is my group now, the current students. Elijah Davis and Marshid. Yeah, that's me over the years. Professional hazards of being in academia.
For the final discussion, yeah, and Orlin, if you don't mind coming up. And can we get Deep, can you hear me still? Or is it this? Hello?
Yep, I can hear you.
Okay, let's see. So we'll kind of open it up to any questions from the audience first. Yeah, go ahead.
I do have a question for Bella. Thank you very much for the presentation. I'm pretty much very interested in the scaling up of the production process for the beginner. And so my question is, how is the circulation that is done? How is it done? And do you recirculate with it? Do you recirculate with the nanoparticles? If you recirculate with nanoparticles, what is the effect of the form on the system during the recirculation? Then in the jar where you have the bulk of the process, where do you have the inclusion? Are you looking at the jar where you have the bulk of the process, or is it at the tip where you have that where you have initially you have the mix of your linear nanoparticles and then the small volume of the particles of the fluid. Then finally, with increasing concentration, I expect my particle size to grow simply because the initial burst of inclusion will result to a large number of particles, and then that will lead to growth by coagulation. And so, how then do you arrest coagulation when you have a high particle concentration in that system?
Okay, can you hear me? Yes. Okay. Well, thank you very much for the question. This is a very interesting question, and it's a very fundamental also. So indeed, what happens is that when you change the concentration, so before the circulation, if you just pass the particle system once, so you are mixing the solution, you're precipitating it. The size is going to be dependent on the concentration of lignin into the solid. The concentration takes the precipitation takes place very quickly. So the moment they mix, they precipitate. So by the time it reaches the vessel, it will have precipitated. And then we recirculate it. And what happens is that we believe that the particles get hydrophilized. So the lignin would not go to the particles that are there already, but would form new nuclei and new particles. And it just keeps adding to the system. So basically, if you follow what happens with the concentration and the particle size in the vessel, the particle size remains the same, but the concentration increases. You cannot do this forever. At some point, you're going to have too many particles, they'll start precipitating. But you can do this for quite some time, and you can increase in this way the concentration, because otherwise you have to work with low concentration of lignin if you want small particles, but then you don't have many in the system, so you need to add. That's how we have intensified the process. Basically, we don't intensify the rate, we intensify the number of cycles that can be performed. But all the action takes place at the moment of mixing, right? By the time they reach the vessel, it's done. We have a paper. I mean, I'll be glad to send it to you, or you can find it out. But we're glad to share that.
The surface area of the particles will control the surface growth, correct? And you're saying if that increases, then you're not going to form a new particle. Is that correct? Am I getting that right?
So basically, every solution that we add to the system is going to form a certain fraction of particles, which are going to have the same size, which depends on the concentration in the solution. Whatever is present there, it is as if it does not exist for the new solution that is added. This is the so-called non-Lammerian mechanism, right? Lammerian mechanism says it will deposit on the particles that are already there, which is usually what happens. But in this case, we have a non-Lamarian mechanism. And this is not that we have not discovered that. This was known before us. But in this case, we found that it works in our system and it works for good because otherwise, when you try to intensify the process, you get larger particles. And when they get too large, they precipitate. Hopefully, can work with other systems as well.
Yeah, go ahead.
My question is for Dr. Mukherjee. In your lasers process, you rotate your metal substrate, right? And then in the picture that you showed, you are kind of pointing your laser to one center. So what is the point of rotating your substrate?
Well, that picture is not it's not the ideal way of doing it because it is you put it at the edge of the thing of your target. And then what it does is that it prevents, basically, you need not do that. It depends on how thick that target is. If you get targets which are very thin, then you don't want to ablate it for too long. With such high energy laser, we're using about 330 millijoules per pulse. It will drill a hole through it very soon for large productions. For small time productions, when we do it, we actually don't rotate it sometimes. We just put it there, and if the target is thick, then it will survive. Otherwise, we rotate it to make sure that it hits a fresh surface so that it doesn't drill a hole through it. It's the only reason. Yeah, go ahead.
Hi, I have a question for the same laser ablation process. I have a couple of questions, but I'll start with why do you prefer liquid based laser ablation compared to the air based that you have done before? And another thing is, what is the time scale of the synthesis? For how long do you have to shine a laser on a particular spot? How long does it take for particle to synthesize? The third question is how you control the size of the particle, and is it when you polydisperse or disperse?
So the first question is why the liquid? So as mentioned that the first ablation thing that I built was for gas phase laser ablation that I had done. And the reason was that in gas phase laser ablation, if I have to make, so we have now gone up to making like ternary nanoalloys, so 3 different metals: platinum, copper, cobalt. Now, such compositions, if I have to do it, which is only gas phase laser ablation, then I have to make an alloy target or a multimeter target and then ablate it. Instead, what I'm driving is that, also the alloy formation will not be that uniform. What I'm doing is that I choose one metal, and then I cascading, I choose the redox gradient. So for example, I have cobalt, and then I have platinum, which has a higher potential, then copper, which has a higher potential, and keep on doing that so that one will reduce the other and will reduce the other. That way, atom by atom, because galvanic displacement reaction will be atom by atom replacement from the particle nanoparticle surfaces. So that was the advantage. One, this is in terms of when I'm making multi-metal intermetallic materials. In terms of, for example, in the case of MOM, the advantage that I got was typically in solution chemistry, it will be, sorry, switch off. Thank you. Typically, it will take quite a bit of time, half an hour for the MOMs to form. In this case, we could make it in minutes because you have these high energy ions coming from the plasma which are being ejected. They immediately go and react with the imidazole and they start the coordination and polymerization to form the MOM. So what I'm trying to say is that the solution, reason of encapsulating the plasma in the solution is that you take advantage of the high reaction rates, the high energy reaction rate of the plasma, and then interface it with the solution chemistry. That way I get immediate chemistry that can happen in the solution phase. And I get the benefit of both. So I can initiate and then form composite materials. Second question, what was the second question again?
What's the time scale? How long?
Yeah, the time scale. So this has been a challenge for us now. That is heavily dependent on the replication rate of the laser. The rate that one that we have now is a 10 Hertz laser, which every 0.1 second there is a laser pulse that's coming. And that's something that we will do with the DURIP that we have. It's an instrumentation development where we are going up to 1 megahertz laser. Why? Because it's basically the seeding nanoparticles that are being formed are the rate controlling reaction. The rate limiting is all the solution chemistry that's happening at the bubble liquid interface. So if I produce a substantial, nucleate a substantial number of particles, and I can create a lot more materials. Right now, we make for Army labs, fortunately their testing requires, the testing that they have developed requires only 20 milligrams. So we can make up to 50 milligrams we have made, and it takes us about 3 to 4 hours. So it's quite a challenging process right now. But what we're trying to do is that if we can go to a very high repetition rate laser, and we intend to increase that by many orders of magnitude, the manufacturing process. That's the large scale manufacturing plan that we have now.
As in the time scale, does it depend on the size of the particle?
Yeah, the size of the particle, as I said, is one of the demerits of the process is that right now we do have a pretty, we get very small sizes, 8 to 10 nanometers, that, as I showed, but there's a large dispersion in it. And the only way to control that we have now is the laser energy, or the fluence, energy density rather. The laser energy per pulse does not play a role. It's the energy density at the spot that plays a role of how much of nucleation, so how much of saturation you're creating, and how much particles are being nucleated. So, and then that determines the size, the critical size of the particles.
One last thing. Is it like a batch process where you want to solution, or is it the continuous process?
No, right now it's a batch reactor. And again, the DURIP will be a continuous flow reactor that we are building so that we can do continuous production. Because we have to deliver approximately 500 grams of these materials to Army lab.
Thank you.
Thank you. Just have one more question on the scale of the system. Definitely, I will take some time to read up the data. But basically, by recirculating, you extend the residence time of the particles in the system. And from your explanation, when the particles nucleate in the system, it's kind of like shielded from the new particles so that they do not interact. And so my question is, why do you then need that big jar down there? Why don't you just, after that small t, when you have the initial nucleation and you have the particles, why don't you just take it out? Why do you need the big jar and the entire situation?
The big jar is needed because, so basically, let's say the company, we have this in my lab just to test, but in the company. So let's say they need to produce a certain volume of lignin nanoparticle suspension, right? So they will put kind of like the volume that they produce in the big jar. So this determines how much will be produced at the end of the process. Because you say, well, I want to have, for example, 1 liter of that much concentration of particles, right? You start with 1 liter and you keep recirculating until your concentration goes up to that level. It cannot go to very high values, right? There are two things. If you go to higher values of a certain concentration, first of all, when you have too many particles, they start aggregating. And second of all, you're adding the organic solvent into the system. So you're going to have accumulation of organic solvent over time. Eventually, you're going to have too much organic solvent. You have to remove it, or you can use it as it is, but you cannot keep doing this forever, right? But you can say, well, I want this much system with that size of the particles and that particle concentration, and you pretty much dial in the parameters and you're done, right? It works very nice. Does this answer your question or?
Yeah, but
Is Deep on the line?
he is still on the line, yeah.
Okay, so that you can hear me.
Yeah, Deep, can you hear?
So the
Yes.
first regarding circularity, I mean, it's an interesting concept. So I presume circularity could be a multi-objective problem, right? So you presented it as, I presume what I picked up is the material going back to the precursor stage. But you could also maybe use your product and make another precursor to make another product and a sequential set of products. Obviously, as I said, it might be an optimization problem. When we deal with other materials, for example, you also downgrade the application. So I make a battery with all kinds of nanomaterials. The battery has a certain lifespan. If it is for electric propulsion in an aircraft like a drone, I need a high quality battery. Then I might use it in an automobile. Ultimately, I might use it in a stationary application. So do you consider such approaches when you look at the circularity problem, or any comments from an industrial perspective on circularity?
So that's a great question, and unfortunately, I can't see the audience, but I think you hit it on the nail on its head. The biggest,
Looking fellow asking the question.
once again,
I said I was a handsome looking guy who asked the question.
I didn't get to see the audience, but that's the essence of what we're trying to do at Eastman right now, is making sure that we are able to process feedstock that is the most challenging in terms of downcycling. So as I mentioned, as you go through this attenuation phase, and you go from clear plastic to colored plastic to opaque material and then to textiles and carpets, there is a clear degradation in the properties of the polymer. What we are trying to do is start off with the more easier feedstocks, which is the plastic bottles and plastic containers. But the real challenge is when we get into processing things like carpet. That's a massive problem in the United States. Carpets basically end up in landfills. There's no way to recycle it today, except putting them in recycles and in landfill areas. So what we're trying to do is take the carpet or the textile from the carpet and then unzip it into the building blocks like DMT and ethylene glycol. So that will define the extent of success we can have with the circular process. So and that's just a slice of the overall material space, because right now focus is on polyesters. But when you look at PVC, polypropylene, polyolefins, the expanse is huge. But to your point is, we can define success on circularity when you're taking the lowest common denominator in the material space as it goes through different cycles and are able to bring it back to its pristine level of performance.
Thank you.
Thank you. Can you ask each other a question?
Yeah, absolutely.
One question. I need the microphone. Well, can you hear me?
Yep.
First was that most of the work that I've seen in terms of bio-based materials, especially lignin biopolymers. Sorry, I'm not from this area. So is that people have worked with lignin. People have tried to extract lignin. I think it's easier to burn off the cellulose. Is there a way to extract the cellulose from natural thing and take away the lignin? And how do we do that? And because the cellulose can have a lot of interesting, especially burn very well. I like things that burn us.
That's a very good question also for our third panelist, maybe. So maybe he can answer after me. So there is a lot of industry that is focused on cellulose, of course. And one of the, I would say, most described, of course, and the most visible is nanocellulose. You have probably heard of nanocellulose being a big hot thing. It has lots of problems, at least from my perspective. Nanocellulose, that is the way it is produced, nanocrystalline. Let me be specific. Nanocrystalline cellulose is more of a problem than a solution, frankly. And it's very expensive. The process is very harsh, environmentally unfriendly. The resulting particles are, well, they may have some environmental problems or even health problems. I'm not a specialist in that, but I am not really enthusiastic about nanocellulose. Something that Eastman probably does a lot is microfibrillated cellulose. And lignin is a byproduct. Typically, you do not make a process for lignin specifically. You have to find a supplier which is going to provide the lignin, which is going to be clean enough to kind of like make a good product. And that's a challenge in its own, because most of the lignin, the so-called craft lignin, is not the cleanest product. It's very inexpensive, but not very clean. One thing that we have done lots of nice research with the so-called HPL, high purity lignin, which was a byproduct of biofuel fabrication. there was at least one company in Canada which was using biofuel enzymatic process to release lignin. It was great lignin, but then somehow they do not produce that much anymore. So finding a good source of lignin, even though it's very cheap, is kind of tricky. And maybe our Eastman colleague can answer further about cellulose. I just want to mention something about also the question here about why we want to recirculate in the first place. One thing about lignin nanoparticles specifically, but maybe other biopolymer particles, is that they are notoriously difficult to concentrate once you disperse them in water. If you try to filter them, they do not filter very well because they're soft, they may plug the filter and they may aggregate. If you try to centrifuge them, they also don't centrifuge very well because they do not have much of a difference in the density with the liquid. So you would rather design a process where you have the final concentration to be the concentration you want, rather than make them at low concentration and then try to concentrate them, just because concentration is a problem. So this hopefully explains some of the motivation of why we want to keep adding until we have the right concentration, right? Because otherwise, there are very well established processes in pinching jet. You can make as much as you like, but concentrating them would be a problem. And maybe our Eastman colleague can tell us about cellulose, which is another interesting polymer, of course. I don't know, Deep, do you have any comment?
Yeah, I actually will have a pretty different view to what was just shared, because for two reasons. One, I spent my graduate work looking at nanocrystals from cellulose. So I'm working in the circular space with polyesters right now, but my graduate research was actually looking at microfibrils of cellulose as well as nanoparticles of cellulose. I don't think it's environmentally unfriendly or it's difficult or more expensive to manufacture. The challenge is, dispersing these particles to a level at which you can get the full benefit of the higher aspect ratio and the surface area. There have been some companies looking at nanocrystals and microfibers for cellulose, and there's been different routes on how you can achieve it. The traditional route was to get the lignin out, bleach it, and then hydrolyze it with sulfuric acid to get the nanoparticles. But I think since then, and this dates about 20 years ago when I was in grad school, there has been significant advances where they are looking at industrial scale production of cellulose microfibers. So these are real, I think, value-added components, especially in personal care and coatings because of the rheology modification. Cellulose has tremendous benefits in how you tailor and optimize rheology if you can make some surface changes. So the full potential, I think, is still not captured, but I'd say there's a lot more runway ahead of us.
I think one more thing that I wanted to, because the earlier question, to your question, to add one more thing that I want to add about the liquid is that I suddenly remembered it because I didn't talk about it at all. I cannot, which is especially for the metastable structures. When you make them, if you have it in gas phase, you'll always make equilibrium structures by the time you collect them. In the liquid phase, what you can do is that from a high temperature, the plasma is at 4,000, 5,000 Kelvin, to a room temperature liquid, it's being quenched instantaneously when the bubble collapses, which happens in about 300 microseconds. So you get it that you have immediately quench it and you can kinetically freeze certain structures, which is something I didn't present, but we have been able to do that. That's another advantage of the liquid thing. I remember.
Which reminds me, there are words on metastable phase synthesis in air, in flame synthesis. We'll talk about it.
Yes, you can do that. Okay, you can capture that, but then you have to actually recently Volkswagen has done it in RF plasma. In RF plasma, they have done it, but then you have to you can only collect it in very small amount. Here we can make up to 50 milligrams, 100 milligrams of materials, of metastable materials.
Okay.
I have one question. I mean, in terms of environmentally with kind of the germ kill aspect of it, in terms of when you have the icosam particle, the kind of imported particle, in terms of the mechanism of the germ kill, I mean, because obviously if there's some sort of kind of range of polyelectrolyte charge density, which kind of impacts it, and you see that in the kind of a range of data potential where you kind of get kind of a maximum in terms of your efficacy, if you will. That's my first question. And the second question is, we can also repeat this, that because you have hydrophobic patches on the particles, and you can put charge on them, have you looked at them as emulsifiers as well from that perspective, or Pickering emulsion?
Thank you. First, I just want to mention that, I mean, I'm not saying that nanocellulose is a bad material, right? It's a hard to commercialize, same as many other natural biomaterials. So nanocellulose is kind of for professor to professor perspective. Nanocrystalline cellulose is the greatest particle to work because, I mean, it really has amazing properties. The commercialization is, again, an issue that is not really kind of like, I mean, something that I can comment too much, but obviously has not been very successful for a variety of reasons. It's still a great material. And microfibrillated cellulose is really the, I think, direction where, at least based on our other collaborations, feedback, industry is going, right? So not nanocrystal in the form of single crystals, but in microfibrillated form. And I think that kind of like, I mean, it's not contrary to what our recent one is. So that's about the cellulose. Now, with regards to the antimicrobial activity, basically, I mean, you can bet that any material that has chitosan at high surface area is going to be antimicrobial. So chitosan is really kind of like a very, I would say, kind of holy grail for antimicrobials because it is, I mean, if you can make it in high surface area, so people put it on circling, on coatings, membranes, I think, nanomaterials. Basically, any material that has high, well, not high, but some positive charge and high surface area is going to be antimicrobial to some extent, at least to most of the microbes. Maybe not 100%, but kind of probably 90% of the microbes would suffer in the presence of high surface area positive.
Right. So my question was,
but chitosan is a bit expensive. Chitosan is a bit expensive on its own.
right.
So if you can use it just as a surface coating, I mean, you're really potentially improving the properties.
Yeah, I guess I was kind of coming at it more from like, if you take like a cationic surfactant and you kind of are kind of adding it to a gram-positive, gram-negative bacteria, the wall is disrupted. So you get attraction due to the ananism of the bacterial wall, but then you get an insertion of the hydrophobic tail as well, which disrupts the kind of the bacterial cell wall. So I'm trying to understand from the chitosan nanoparticle perspective, the charge binding takes place, but there is a subsequent disruption of the wall due to hydrophobic patches kind of disrupting it.
That's a very good question. I would say that, well, this is what you answer when you're not sure about the kind of like what would be the exact answer. Right? Even at least some years ago, even the antimicrobial action of silver was not controversial, but there were many hypotheses and kind of like it turns the bacteria disrupted in many ways. Definitely, high surface area positive material would be antimicrobial. Of course, cationic surfactants are very well known antimicrobials, but then they have a number of potential problems in terms of toxicity, environmental, and so on. So I would say that chitosan may not be as efficient as a cationic surfactant, but it is considered to be safe. So that's why people are interested in chitosan. On the other hand, chitosan is, again, like the issue of chitosan is the price. That is the cost. It depends on the purity. There are interesting developments where companies try to make chitosan by basically growing bacteria in bioreactor. So this is an alternative, right? Not by processing, but by biosynthesis of chitosan. I'm not sure how well that is going, but kind of like an interesting development. But it's a good material, as I mentioned. It's kind of like I consider it to be the high-tech biopolymer, right? I mean, it has very good properties. Yeah, we're making fumes out of chitosan, outstanding mechanical properties. Right? A fume out of chitosan can be better than a polymer film, but it won't be immediately usable because of the price.
Yeah. And my second question was on the emulsifier, and when you look at it,
Yeah, there are lots of people who do emulsification with lignin. It really depends on what you want to emulsify. So most of the emulsification that people use lignin for emulsification, industrial emulsification, of kind of like really inexpensive products. The lignin that is used, and this is kind of important difference to make. There is a so-called sulfur lignin, which is basically physically it's a different material than the lignin particles that kind of would be really lignin. Right. So sulfur lignin is basically a surfactant based on lignin, and it is used in inexpensive products, which I am not sure how well they're going to be like in terms of CCP and other applications. Making pure lignin particles and making clean, potentially non-toxic Pickering foams and emulsions, I think that's a great topic. Right. This is right now cutting edge, right? I mean, people are trying to do that. If you're working on this, good topic.
Right. Okay. Any other questions? I know we're kind of at time now. We just hit 4 o'clock. So if there's no other questions, I'd like to end the session. I don't know, Pratik, if you have some parting words.
Yeah. I think thanks firstly to all the speakers and thank you all for hanging on till the end. I hope you had a good workshop. We intend to do this next year too because we intend to do it next year too because we guarantee the excellent weather. Okay, and so please come back. We will do a survey. Please let us know because we are also tweaking the first one. So on behalf of all the past members, I must also thank all our students, staff who put in some effort to put this thing together. Thank you very much. And of course, thank you to all the attendees. And once again, thanks to the sponsors, Hendrix, TSI, and the third one is of course Safe Cooper. So thanks very much. Okay, and have a safe trip back. I think there is a visit to the laboratory, so Shalini will coordinate that. She's right at the back. Whoever is interested, please make the most of it.
Yeah, we'll go from here only.
Yeah. Okay. Thank you. That would conclude this workshop.
Good night.